will have this polyglutamine tail at Huntington. So if this polyglutamine tail is too big, then it is problem for the removal or degradation of the protein. The protein will be not anymore removed and it will start to aggregate. So from the number of these repeats, it depends whether it is pathogenic or not. So if there is less than 29 CAG repeats, then it's always non-pathogenic. If it's from 29 to 35, then it's sometimes rarely pathogenic. But then after that, the probability starts to increase. So if it's 36 repeats, then probability to get Huntington disease is 25% and so on, 50%, 75, 90. And if it's more than 39, then it's leading always to Huntington disease. So it's very strict. One repeat more and risk is going up. So this is the best known such kind of polyglutamine disease. There are also more or less frequent polyglutamine diseases. So where this polyglutamine is not attached to Huntington, but to some kind of other protein, which leads to different disease. So, for example, as ataxias, mild motor neuron diseases, and so on. So, okay, so mutant Huntington aggregates. What happens because of that aggregation? So, a lot of different things. So, first, that gene expression is affected. And the second, protein quality control is somehow affected. Mitochondrial function is affected. Axonal transport is affected. Synaptic signaling is affected. The reason why everything, quite many things are affected is some of them are more primary, some are more secondary, is that Huntington has quite many different functions, not fully clear what it is doing, but it is involved in many cellular processes. So I'm just showing what it is doing in axonal transport, so that this Huntington is also interacting with motor complexes. So you remember from the yesterday, then this anterograde transport was coordinated by kinesin and retrograde transport by dynein. And the Huntington and Huntington associated protein are also interacting with these complexes. Don't know exactly what they are doing there, but they are regulating or affecting these complexes. And if you take mutant Huntington, this mutant Huntington will spoil these movements. So anterograde and retrograde transports both will decrease. And if transport is decreasing, then everything from mitochondria to synaptic proteins will start to move more slowly. Synaptic terminals receive less essential components, synapses don't work properly, and this leads to the problems. So if you take the guidelines, then in guidelines, in Europe you have really only one drug, tetrabenazine. In the US you have amantadine and riluzole as well. So what is the logic why they are used? So one thing what is the main problem is that patient have this involuntary movement. So the idea behind this drug is that it will be good or necessary to block this involuntary movement. So the tetrabenazine blocks vesicular monoamine transporter, reducing the uptake of dopamine. And if you have less dopamine, you have less movement, as you remember from the Parkinson's disease lecture. But it's only symptomatic treatment. It's effective at the beginning. You can see the graph here that this is the involuntary movement, and the involuntary movement is not progressing so quickly if patients are treated with tetrabenazine. It has quite serious side effects, so it is making depression more serious, and it is also inducing other psychiatric conditions, including the suicidal thoughts. Then another drug, I don't speak about the amantadine and riluzole. Another drug that is maybe coming is pridopidine. So the first clinical studies showed that there is some improvement in motor function, but this was not enough to get the regulatory approval. And one of the first studies also showed that this primary endpoint, that this is slowing disease progression, was not achieved. But secondary endpoints were achieved, and the company behind this drug is developing it further. So what is the point? The point is that it is sigma-1 receptor agonist. Sigma-1 receptor is in ER membrane. You can see here it interacts with IP3 receptor. The mechanism is not fully clear, but the idea is to modulate ER stress, to modulate ER function, to modulate ER mitochondria contacts. So the function of the protein is not, receptor is not fully clear, but it is involved in these processes. And this is somehow, through the calcium flux, is affecting the aggregation of the huntingtin. So the sigma receptor agonists are now, they have been studied long time ago, but now quite many of them are in pipeline for different diseases. And if I remember correctly, this pridopidine is one of the drug candidates which has reached to this, which one of the first drug candidates which reached to clinical trials and are close to something. So, any questions about Huntington disease? So it's different from others. There is only one gene and only one type of mutations. So theoretically it is most easier to find target for the treatments. In case of Alzheimer, you don't know what is the target exactly, what is the primary reason. Here you know that. But okay, any questions? Then we will go on. We have still some diseases left. So amyotrophic lateral sclerosis, motor neuron diseases. So this amyotrophic lateral sclerosis, ALS, in States they use also name Lou Gehrig disease. And the reason is that it was first described or acknowledged because of Lou Gehrig, who was the US baseball player. So it was in 20s and 30s And he was a very good player, but then what happened that within one season his performance suddenly and very quickly become worse. And people were, baseball was very popular then, probably it's still popular. People were worried, and then they understood that this is this ALS. So it was acknowledged, I think, because of Lou Gehrig, and disease got there the name according to that. And one of the most famous patients in modern times is Stephen how to pronounce it, Hawking. He was diagnosed first with ALS at the age of 21 when he just finished the university. And since the life expectancy in case of ALS is relatively short, and it was short even then, so it was just a few years left, and there was no hope to do anything. But what really happened that when he died at the age of 76, then he was one of the most famous physicists in the world. So despite of diagnosis, he managed to do a lot. So what this tells that although the life expectancy is very short, it's not one disease, it's a spectrum of diseases, and in some cases 5 to 10% this disease progression is very slow. Okay, so ALS is just one of the motor neuron diseases. So the motor neuron diseases, as the name says, they are affecting motor neurons. And we have in body two kind of motor neurons. We have these upper motor neurons. They are in brain stem, and then we have lower motor neurons. So they are in spinal cord. They are also in brain stem. I mixed that. So upper motor neurons and lower motor neurons. And we have this diseases, specific diseases affecting the upper or lower motor neurons. So for example, if we will start from the figure, we have these upper motor neurons diseases. They will lead to primary lateral sclerosis. Then we have a PMA, which is progressive muscular atrophy. This is related to lower motor neurons. And then we have a bulbar onset motor neuron disease, which means that mainly the brain and spinal cord neurons will be lost. And best known is the ALS itself, which involves actually both upper and lower motor neurons. What really then happens? So in any case, whatever it's upper or lower motor neuron, the diseases have one main cause. The motor neuron axons, also the full motor neurons, they will die. And the motor neurons, they are cells sending signals from brain to muscles, or from spinal cord to muscles. And if they will send the signal, then muscle will contract, and that's it. So if you don't have any more the motor neuron axon, then the muscles cannot anymore contract. They are not anymore innervated, they are inactive. So this is one thing, that you don't have movement, but if your muscles don't anymore contract, they will start to shrink. So this means that there is atrophy. So if it goes on, all muscle fibers are denervated, this muscle will go away, to say it simply. And this is the main reason. First you can't move, then you can't move at all, and then you can't attend, you can't respire anymore. So you will die because of the respiration problems. What is the reason? As Alzheimer and Parkinson, it is either sporadic or it's familial. Sporadic forms, they're most frequent, 90%, familiar 10%. See, these familial forms are related to different genes doing very much different things. The first gene or first mutations found, they were in SOD1 enzyme. This SOD1 is superoxide dismutase, which is neutralizing the free radicals. So, but if you take all these proteins affected or mutated together, then again, as in case of previous disease, you will see that you have problems everywhere. So it's, if we start from here, then there is dysregulated vesicle transport. Some of the proteins involved are related to vesicles, are related to autophagy. If you take, for example, this optineurin, then it's the protein homeostasis, ER stress, DNA damage repair. Most well-known is RNA problems in RNA metabolism, mutations in genes like this TARDP and FAS. They will disrupt RNA processing, transport and splicing, and this leads to toxic cytoplasmic aggregates of this TPD43, which is the hallmark of the ALS. So around 90+% of the cases have this TPD43 aggregates. And these aggregates are bad, they are toxic because they sequester RNA binding proteins and impede RNA metabolism. So just to remember, many mechanisms, but this TPD43 aggregation leading to RNA metabolism problems, this is one of the most important. And then inflammation, mitochondrial dysfunction. This first gene SOD was mitochondrial, glial dysfunction, and probably as a secondary, or maybe not so secondary, oxidative stress and excitotoxicity. So the drugs available, they are targeting oxidative stress and excitotoxicity. So Riluzole, approved 30 years ago, it is inhibiting glutamatergic signal transmission. It is blocking the excitotoxicity. People were thinking that also other glutamate signaling inhibitors are protective in case of Huntington, but in reality they all failed in clinical trials. So the reason behind that is that Riluzole is not just glutamate, receptor antagonist, but it is doing many different things. It inhibits sodium channels, it activates potassium channels, inhibits calcium channels. So it inhibits this glutamatergic transmission in many different places. Maybe inhibition of the sodium channels is most important. it's not so efficient at all. So if you take this Kaplan-Meier survival curve, just to remember, this patient survival curves are frequently called Kaplan-Meier curves. So you have here the survival probability from 0, that is good, down to 0, that is bad. Then you have placebo, patients are dying, and then you have a riluzole, patients are dying, but maybe not so early. So it's only additional 3 months to life, not really very much. Then another drug is edavarone. Edavarone is antioxidant. It's unfortunately administered intravenously. It's relatively new, almost less than 20 years old. If you take again this survival curve, then it looks slightly better, but you add maybe, 20 months to life, and it's unclear what happens at the end, but it seems to be from this curve that it is more efficient. So what is maybe also important is that it's not working in all patient groups similarly. So in some groups, specific groups of ALS patients, it's more efficient, and more modest or less, it's less efficient in some other patients. Then it's interesting, one new drug that came and went. So for me it's especially interesting because it affects the ER and mitochondria both, and I knew how it started. So it was 2022 when FDA approved the combination of the sodium phenylbutyrate and taurosodiol. So the price for that drug was quite huge, more than 150,000 euros per year. But these original compounds used here, they were already in market before, and they are very cheap compounds. So actually the price tag came from nothing. You can understand why the gene therapies or the antibodies are so expensive, but for this very simple compound combination, the price is just coming because it's difficult to develop everything and so on. So it was considered at the beginning such kind of first disease-modifying treatment. So how it was first approved was also unusual. So normally FDA wants to have 2 clinical trials, at least 1 phase 3 trial to approve new drug. But if the disease is very serious and there is no really effective treatment, then they can accept 1 trial also and some supporting data. So this specific drug had only this single phase 2 trial with 100 and something patients, and it was really very small clinical trial, and at the beginning FDA said it's not enough to approve it. This small trial shows nothing. But then the patient groups were becoming aggressive and strongly pushed it further, and then FDA, because of that, even without strong evidence, approved it. But what then happened is that when it was approved, and the condition was that they performed this phase 3 trial, but what then happened that this phase 3 trial was negative. So what happened then is that nobody is anymore using it very much, and the stock price of company went significantly down at that same day. But what is interesting, what is the logic behind that drug? So it was the first compound was the inhibitor of unfolded protein response that reduces also ER stress and ER dependent problems. And then another was mitochondrial or PAX inhibitor that reduces mitochondrial dependent death autophagy. So it's the ER stress and apoptosis inhibitor together. So they are not very specific compounds, especially this PB, but they are still doing it. And just back to this initial study, the initial study showed relatively small difference between groups. This red is the drug, it was slightly better, but okay. So any questions regarding the ILS? It is not, as you remember from first slides, it's not very frequent. It is, but it is quite, how to say, I know at least few people who got the diagnosis, and after that they had only few years left before they died. So it's quite fast disease, and it's quite a nasty disease. But okay, let's go on and have a discussion at the end. So the spinal muscular atrophy, SMA. Yes, please.
So this PB and also drug, so they target UPR response.
Yes.
So can this drug can be used for Huntington patient, because they also UPR response is also there.
I don't know. I think the company is currently not studying that. This company now tries to use it for Wolfram syndrome, where there is strong ER stress. But I think in case of Huntington, the ER stress is not so big problem. So I think it's not. It is, but I think it is there quite secondary. But okay, I agree that it might affect the calcium fluxes and Huntington aggregation and so on in cytosol. So maybe there is point in that. But for whatever reason, I think they are not doing that at the moment. At least not. There is no public information about that.
You have mentioned the TDP-43. Could it be a possible therapeutic treatment for the Alzheimer? TDP-43.
Well, theoretically, it's possible to treat, but I think they have no compounds at the moment for that. But I don't know that part very well. Some more questions? So this spinal muscular atrophy, SMA. So it's most it's quite frequent, and it has 4 subtypes. The subtypes 1, 2, 3, 4, they are this classification is based on the age of the disease onset. So in case of this first type, SMA1, it starts very early and life expectancy is usually few years. Type 2, this means that it starts later. Type 3, it starts in childhood, even later. And type 4 starts usually at adulthood, and it's not affecting really the life expectancy. Type 3 is so-so, it starts relatively early, but life expectancy is not so considerably affected. And the reason for that is that motor neurons are dying. They are dying because their gene, which is called this survival motor neuron gene, SMN, is having problems. This important survival motor neuron, what is the name of that protein now? I forgot it fully, sorry. It's not expressed anymore, and neurons have problems. So it's SMN, survival motor neuron protein, I think is the name, yes. So this survival motor neuron protein has in a cell many different functions. So mRNA splicing, so different functions. One of the functions is again the axonal transport that is affected. So whatever is the exact mechanism, it's clear that this protein is needed for motor neuron survival, and without that motor neuron will die, the axons will die. Could be axonal transport, that is one important thing, but not only one. What makes this disease different and important is that it has quite okay therapy. So, and it's a gene therapy. It's one gene, and it is loss of function, which means it's the easiest situation for gene therapy. So before this gene therapy came, this Zolgensma, this gene therapy is just the viral vector that encodes the SMN1 protein. It goes to motor neurons will start to express this survival motor neuron protein, and motor neurons will survive. Before that, there were also 2 other gene therapies. Not exactly gene therapies, but therapies affecting genes. Both were related with that there is, in addition to this SMN1, there is another similar gene, SMN2, that encodes slightly different protein. And one of the therapies changed just alternative splicing of this SMN2 so that it was becoming more similar to SMN1 and compensated partly. But these antisenses and compounds, they were not so good because you had to administer them all the time. Expensive therapy, lifetime therapy to get this alternative splicing all the time. But then came this Zolgensma, which is this viral vector, and it is the first one, if I remember it correctly. and this was really efficient. So if you take again this survival curve, one is everybody happy, then this gray line means the normal situation. Most of the 80% of patients or 75% of patients die within 1 year, but with this viral vector treatment, the patients survived and only 10% died. And this is also not just because of the inefficiency of treatment, but usually the treatment follows the diagnosis. And if diagnosis is coming, then sometimes already it's quite late. A lot of motor neurons are dead, and it's too late. If there is no motor neuron left, then the therapy is not helping. You should treat with this vector when you still have a motor neurons there. So early diagnosis is really most important. And why for me it's interesting is that one of the Estonian patients, it was many years ago, one of the Estonian patients, when the therapy came, was sent to America to get this therapy. And there was a lot of information about that in newspaper. And I also have some, how to say, private information from the involved medical doctors telling how the treatment was continued in Estonia. That it's not just that you will administer the vector in States and everything is okay after that, but you may have some complications because you will have not one copy of this gene in motor neuron, you may have several copies, and you have some secondary effects and side effects that vary from patients to patient that should be, how to say, considered and treated. And at that time it was the most expensive drug, single dose, you don't need more than single dose, was $2 million. Any questions about that? So we will have discussion then at the end. So prion diseases. So this is just interesting. So they are most aggressive, if we could say, neurodegenerative diseases or disorders. And they are characterized by the accumulation of the abnormally folded prion proteins. And it has this group of diseases is quite heterogeneous and has many names. So the most well-known name is Creutzfeldt-Jakob disease, sporadic. Then it could be also the Gerstmann-Strauss syndrome, fatal familial insomnia, which is, as the name said, familial. Then earlier, when it was in Papua New Guinea, it was called Kuru. Now there is no Kuru much anymore. Then it was in sheeps, or it is in sheeps. It's called Scrapie, and Bes in cattle. So in all cases, the primary symptom is dementia in humans, and there will be motor dysfunction, ataxias, and a lot of other things as well. So it's interesting how it was discovered. So it was discovered actually as this Kuru. So it is disease that once was found in this Papua New Guinea islands, in some islands. And the local population they were thinking that it's some kind of sorcery, and the white people were thinking that it's such kind of psychosomatic reaction related to Western colonization. So actually people had no idea what is behind of that disease. And then in late 50s, Dr. Kadiuszek showed that this is actually associated with cannibalism. And he got the Nobel Prize '97 for that discovery. And this research was quite different what we are doing in the lab. He just went to this island and was living there with locals. So at that time, in these islands, they had a lot of different tribes and clans, and these tribes and villages, they had constant conflicts and wars with each other. They were killing each other all the time. And what was the habit that if you killed a warrior from another village or tribe, you were eating its heart fresh, and only warriors were eating the hearts of these other warriors, not children or not women. So what happened after that? It was that people were dying, and only males were dying. So when the males were young, everything was okay. When they started as warriors, then a few years later they died. So it's just a graph from the Kadiuszek publication showing the number of male deaths because of Guru, the female deaths, and how the male deaths decreased. Also female deaths, but how male deaths decreased during this period. So he also showed what is behind this, that in brain there are a lot of intraneuronal vacuoles in Purkinje cells, and that the brain at the end is looking like this sponge, and there is atrophy. And that there is some kind of protein, prion protein behind that. What is maybe more important is that when there, he convinced the locals that killing and eating other people fresh is not good. And they really understood that this Kuru disease is because of that, and they stopped this cannibalism because of him. So he had a lecture, I think 20 years ago in our university, and it was really fascinating to see all these images. But okay, what this protein is doing, why it's different from alpha-synuclein or beta-amyloid, this is that it aggregates more aggressively. So how exactly it aggregates is not so clear, but you have a normal protein, everything is okay, its function is not fully clear, but somehow the normal protein is becoming not normal and starts to accumulate. And these big accumulates are everywhere, they're spoiling the function, neuronal function. There are two options or speculations how this happens, but most prevalent is that these bad conformations will interact with, in dimer or in oligomer, will interact with normal prion proteins, and they will turn in these dimers the normal prion protein to bad one. So then you have two bad proteins, and they will go again and turn the good ones to bad ones. So that what Kadiuszek showed and others, that in principle you need only one bad molecule, and since it's infectious, it could turn all other ones to be bad. So in reality you need more, that since the probability, if you eat it, for example, the probability that this specific bad molecule will reach to brain is not so high, but theoretically one molecule is enough. Then this is infectious version. So there was maybe, don't remember, 10 to 20 years ago, there was a scandal in Europe where they had this Creutzfeldt-Jakob disease in UK, less in Europe, mostly in UK. Not too many cases, but much more than usual. Usually is very low. And the reason for that was that if we go back to this figure, slide, that the sheeps had a scrapie, they had this prion disease. Then the sheeps were used to make food for the cattle, so that the cattle were eating some kind of food made from the sheep, and even if it was processed, this protein was stable enough to be resistant to that processing. And then the cattle become infected, they got this PSC, and people were eating that meat, and usually in Europe beef is eaten quite, it's not heavily processed usually, people eat it, and sometimes it's quite raw. And through that scheme, some people got these prion proteins from sheep, and it was enough to get the prion disease. This is one possibility. Another is familiar, so that there is this, fatal insomnia, which means that some families, very rare in France, have mutations which increase the probability that this protein will turn to this bad conformation. And very rarely it could be sporadic, that this just happens without infection and without mutation. So mostly anyway, if you take worldwide, it's mostly sporadic, it's very rarely infection, and then in some places you have mutations also, so genetic component. So why it's interesting is that it's infectious. You have one bad conformation and that's enough. So if you take, for example, alpha-synuclein, then one theory is also that it is little bit infectious, that you have alpha-synuclein, that it is going like prion, not so aggressively, but it's going to other neurons, and it will also infect other synuclein molecules in these other neurons, and it will start to accumulate. So this prion theory is relevant to understand other neurodegenerative diseases as well. So, a few questions about before discussion, or you have some questions or not? Then we will go to discussion.
Just
Yes.
out of curiosity, what could happen while treating the heart? I mean, what was actually going into the brain which caused the aggregation of the proteins in the Guru disease?
In Guru disease. So this protein is expressed everywhere, but the symptoms are related mostly to the brain. So I mean that in brain it accumulates and induces such kind of problems, but it's also everywhere in the body in lesser amounts. And since it's infectious, you will get enough load if you eat heart. So there is no need to eat the brain only. That is my understanding. So, questions, very general questions for you. So this first question I asked already at the beginning. So why the neurons are very vulnerable? So we had a number of different reasons for that. Then question to you that, okay, all these diseases were different, but what is the common mechanisms? If you can think about it, what can you find something which is absolutely common for these diseases?
Mitochondrial dysfunction.
Yes, oops. What do you think, how common it is?
So it occurs in every disease in the organism. Like mitochondrial dysfunction, neuroinflammation.
Okay, what else? Sorry? Oxidative stress, which is coming from what? Where Ross is. So it's also mitochondria, a little bit mitochondria related. What else? It's not sure. Sorry? Protein misfolding. It's again, it's like mitochondrial dysfunction. It's not 100%. You can find some neurodegenerative diseases where nothing is folded wrongly. But it's in many cases very important. Exactly. Why do you think it's, is it secondary or primary? I mean, I think it's in most cases secondary because something is going wrong with calcium, with transport, and then it's excitotoxicity. Something else? Because what you mentioned, excitotoxicity, so many drugs we covered, they were blocking the excitotoxicity. Some drugs we covered, they were antioxidants. So we have no really mitochondrial drugs because just we don't have mitochondrial enhancers, MitoQ, but it's not really drug. What else we can have? Protein folding problems, so we can't really modify that generally. Then this stress, UPR, this we can maybe some kind of stress inhibitors, we can modify it a little bit. What else is very relevant? At least one thing which is relevant you have not yet mentioned. Yes, this is relevant, yes, everywhere. It's axonal transport, because in many cases the axons die, and axons die because of transport problems. And again, if you think how you modify that, you can't really modify that very well. You can, of course, little bit change the anterograde, retrograde transport. Because in case of what we are studying, in case of Huntington diseases, for example, one thing is that all transport is inhibited, but if, for example, anterograde transport is inhibited less than retrograde, then this means that all this stuff becomes accumulated at the end of axon, which is also bad. In that case, it's dysbalance of axonal transport, but okay. So I think we covered most of the things. Then, why it's difficult to have early diagnosis? So it's such kind of philosophical question, because we don't have biomarkers. and we can take biopsies from the brain. Imaging shows only what happens later. Okay, in Parkinson this may be working, but in other cases you can get the early diagnosis. And it's biomarkers. Lack of biomarkers, lack of specific biomarkers is a problem. So a lot of money is going to biomarker development. Even in some cases when you don't have a treatment yet, people are thinking that treatment is coming at some point and we need a biomarker anyway. So again, more philosophical question. So the protein aggregation is such kind of central thing. It's not always there, but in most of cases it's there. Is it primary or secondary? In case of, if you take this prion disease, it's clearly primary. But if you take, for example, let's take Parkinson's alpha-synuclein, is it primary or secondary? I don't know. So it's not so clear. Is it? Maybe there is something else which is doing, and which is doing something, and this is quite secondary. Who knows? Okay, then therapeutics. So most of the drugs we covered, they were treating symptoms, not causes. Why we don't have disease-modifying therapies? Just few. In case of MS, we had many. In other cases, almost nothing. Yes. So it's actually the main reason is that we don't know the mechanisms. Okay, we know in some cases the mechanisms are very different also, and then it's the problem that all these subgroups have different primary targets, and it's difficult to find a drug for every target, or it's not practical. But in most of cases, if you take the Alzheimer, sporadic Parkinson, we actually don't know what is this initial reason. Okay, synuclein is aggregating. Why it's aggregating? We don't know. A lot of speculations. If we don't know, there is no hope to think about drug. So only thing we can do, we can try to find something which is decreasing the aggregation. But what is inducing the aggregation, we don't know. We don't have enough basic knowledge. Then what do you think, can we affect something with lifestyle interventions? Exercise, training. How much we can do that? Can we, or I'm better asking, can we improve the symptoms, or can we change the course of the disease with lifestyle interventions? Okay, so I fully agree that you can affect the symptoms. So if you are more active, mentally active, physically active, then you can suppress the symptoms of Alzheimer, I'm sure. But I'm not sure whether you can modify the disease progression. I think if you take alpha-synuclein or beta-amyloid, they are still aggregating. They are still inducing the problems, inflammation, etc. You can't do very much with physical activity there. And diet, I think also with diet you can't do too much. So I think it's mostly lifestyle interventions will will help with symptoms, but they don't modify really the cause of the disease. It's my subjective decision. And then last thing is the gene therapy. So we had one gene therapy case. What do you think? Is it possible to have a gene therapy against Alzheimer and Parkinson? Why?
Because multiple mechanisms, cholinergic dysfunction, inflammation, oxidative stress, microbiota hypothesis, so everything cannot be targeted by targeting the gene of the interest.
But all these, what do you think, if you take again the Parkinson that I know better, so you have many different hypotheses in case of Alzheimer as well, and you could have many different initial factors. They will give the same symptoms. What do you think? Do you have some kind of common protein which could be effective? For example, if you will have a gene therapy suppressing beta amyloid in general, or gene therapy suppressing the synuclein in general, or gene therapy making synuclein different so it will not aggregate. You have synuclein, but it is not aggregating anymore. So then it doesn't matter that you have some problems in gut or in mitophagy or somewhere if its synuclein is not aggregating.
Sir, there has been multiple studies that are targeting beta secretase, which is one of the primary enzymes that is cleaving the amyloid precursor protein and leading to formation of amyloid beta. But there has been no success in that. Their base inhibitors are researched a lot, but there has been no success in that field.
Okay.
So that is.
Yeah, I agree that you can theoretically maybe consider it possible, but most likely it's not possible against the sporadic forms of these diseases. But then if you take the forms, familial forms of Parkinson disease, what do you think? Is it possible to have gene therapy in that case? Let's take this early onset Parkinson mutations related disease. Can you have a gene therapy in that case? Theoretically. Okay. So then you have recessive and dominant negative mutations. Is it if we will take this BINC1 mutations, then it's recessive. This means that you will just overexpress or you will introduce normal BINC1 and you have BINC1 and it will start to work. But if it's dominant negative disease, can you then do the gene therapy? Because you will, if you will send normal gene in, you have this not normal still that suppresses it. What do you should do then? Let's just think this mutant Huntington as an example. Can we have a gene therapy in that case? Theoretically, again, it's possible. No, it's. Yes, but theoretically, you can first suppress the endogenous expression and then introduce the normal one, or you can correct the mutation in Huntington. So theoretically it's quite possible. So it's different. The gene therapy is not only that you introduce new gene, it could be more complicated, that you are correcting the mistake or you are suppressing the problems and you introduce the new one. Okay, just to be able to summarize what I know. and what we discussed, that if it's sporadic neurodegenerative disease, then it's really complicated to use gene therapy. Theoretically, it's possible that you introduce something more generic, like you overexpress some growth factor and so on. If it's loss of function, recessive mutation, then it is working theoretically quite okay. And you have this example, this Zolgensma in case of SMA, and there are also some other examples. They are not just all virus-based. There are also some other options. As you see, okay, we don't go to diseases, but it's this autologous HSC transplant, and what else we have? Intrathecalized, so delivery, which actually means, just to explain you, that something will be injected to spine so that it will reach to CSF, or the patient's cells will be taken from the bone marrow, they will be modified and introduced back. In both cases, this specific disease is related to glia deficiencies. So some cells you will put back will go to brain and will start to produce the enzyme which is missing there. So that if it's loss of function, then you have different ways to get that function back. If it is gain of function, dominant, then it's more complicated. You have to stop the endogenous expression, and then sometimes it's not enough. You have to introduce the normal gene back, or you have to correct it. So if you take this SOD1 ALS, related ALS, then it's fully gain of function. It's not just dominant, but it's gain of function, which means that you really need to block this bad SOD1. You don't need to introduce back the normal one. So if you take the Huntington disease, then you need Huntington protein. You should silence it, but you probably should send it back. So actually everything is possible if you know exactly the mistake, where is the mistake, and if the mistake is genetic. If mistake is not genetic, then it's more complicated. And what is missing from here, PINK1 is missing for here, but Parkin is missing for here, but I know that this Fox Foundation is supporting currently the study aiming to develop the gene therapy for this early onset Parkinson. Okay, that is all from me today. Thank you.
We have a break for some time, and then we'll get it back. So, what we do is like download sleeves. So, I asked my students
Okay.
after they will show you around the university.
Okay.
Now, the Gandhi Bhavan will be open and our library will be open. So, they can take you to the library.
Okay, that's good.
Yes. And Gunajot will do it. It's okay.
Yeah, it's okay. And then after that, it will be good to have time to prepare for tomorrow.
All right. If you want, then you can go back to work.
Yeah, because I need more slides.
What about lunch? Do you want to have lunch?
No, I think no. I will have something in hotel. It's easier, faster.
Okay, fine.
Yes.
So then,
Yes.
after the university round, then
Yeah, good.
I'll ask him.
You know how many minutes we had yesterday?
Oh, I asked him, even I don't know.
Uh-huh.
So he said, don't worry about
We
much
should not
about
worry.
it.
Okay.
It's
Because,
not,
yes,
you just have to make it. I mean, 15, 20 minutes here and there doesn't matter.
Okay, because yesterday he said it's very important to have exactly, and okay, good. Okay.
Even I was not aware, so I just asked him. I said, we maybe have to make this much better. So he said, it's okay. 15, 20 minutes
Okay.
We just
Okay.
have to, on an average, we have to
Okay.
just
Good.
take the
And then tomorrow
one.
I have lecture and tutorial.
Yes.
So I have a little bit. Also, I think half of the.
You might be tired
No,
standing.
it's okay. Also, half of this tutorial, I think I will, since it was slow yesterday, I will make a lecture longer a little bit and try to have lecture as a discussion or maybe we will see. And then one more question is that Friday there is tutorial which was the poster. So there will be some posters.
Yeah,
Or?
I want to discuss that with you. So I was planning that whatever we teach them,
Yes.
we'll ask them to make some presentation, not presentation, one poster, e-poster,
Uh-huh.
okay? And they will, since we have 35 students, so we can divide them in a group of 3.
Uh-huh.
So we'll have around 10 to 11
Okay.
posters.
Mm-hmm.
So they will present it for 5 to 7 minutes.
Okay.
So that poster should be, I was thinking it should be like what they learn in this course.
I see.
Maybe we can discuss about it. This is what I was thinking, that maybe we can give them that find any neurodegenerative disorder.
Mm-hmm.
What would be a target, therapeutic target in that,
Okay.
and what drugs are available, what are the
Uh-huh.
drawbacks, what are the
Just I had. Okay, just not to make it too. Oh, sorry, I have to take. Because I thought tomorrow I have a plan that I will speak maybe 3 hours or 2.5, and then we will do a target profile, which is just this, more or less like that. What is minimal target for the new drug development? And I will give them for groups, I will give some rare neurodegenerative diseases, not Alzheimer's, but something very rare. So they have to search, they have different diseases, they will go to the table, they will correct and then they will send it to the. It's a little bit similar to what we proposed for Friday. But I will unfold. This is quite specific, so it's target for. Yes, yes. So I will give them disease, but they have feeling that they. Okay. Yes, but the disease, they should find about disease, what is the drug, what is the minimum requirements for new drug, and so on. Yes, for group. But rare one, not the one we covered here, but something which is very specific. So I think it's slightly different. Okay, good. Yeah, yeah. So it's a different. Okay. Then if you could also tell that tomorrow they should take computers. Yes. They don't have it or. Okay, they need internet. There's something.
plant and your book in my car.
I saw
I
it.
forgot. So today I need to
Okay, okay. Don't worry. Yeah. Don't worry so much, you enjoy it, but not too long, because I'm still worried about the notebook's presentation. It's only half done.
will drop him back to the hotel. So tea आ गई है easily. So we want samosa on this one.
I can take the tea, but I don't want to eat. So that's
Same
that
like
in the
okay,
morning.
thank you. Yeah, चाय नहीं है बाजू में ये अपने कॉलर पे लगेगा.
लेक्चर स्टैंड में अगर आप मूव नहीं करो तो लेक्चर स्टैंड में ये माइक यूज़ कर लो.
अच्छा, दोनों ही कर लेते हैं.
यहाँ से
ओके. हेलो, है ना? ठीक है. थैंक यू. So we can start. So now this topic is navigating drug development to target endoplasmic reticulum in neurodegenerative diseases. So I am Dr. Tanzeer, I am coordinator of this GIAN course. So now what is the journey of a drug development? How does a drug development starts? So there are 5 stages for drug development. The first is that first of all we should know about the disease. We should know its molecular mechanism, and from that molecular mechanism we find out the targets. So the first step of any drug development is finding a target, selecting a target. So after we select a target, and it's not easy, as Alan has shown you so many molecular mechanisms of diseases, now you know that it's so complex. Many diseases are so complex. We don't have a single target in one particular disease. That is why we don't have cures for neurological disorders, we don't have cures for cancer, because the mechanism is complex. But as we will do this basic research, as we delve deeper in the molecular mechanism of these diseases, we may come up with some targets and then design drugs against them. So the first step is selecting a target for which we need to develop a drug. Then second step is lead discovery. Now you know a target. Your target can be an enzyme, it can be a receptor, it can be a protein, it can be a DNA. On the basis of, it can be a carbohydrate, it can be anything. On the basis of the type of molecule that target is, you select a molecule, a lead molecule which could bind it, either enhance its property or decrease its property. So the second step is lead identification or lead discovery, finding a molecule which can modify the target and thus impact the disease progress. Then comes the third step in drug designing, that is medicinal chemistry. Now we can find any lead molecule which could modify your drug target. Just a minute, एक मिनट आप इधर से आना पीछे से. So we have found a lead molecule which could modify that particular target, but then we see that how much effective is that lead molecule, and then we work on improving its efficacy, improving its properties. It should be good. I mean, if we give this particular molecule to a human being, it should have a very good absorbance. It should actually reach the site of target, and it should have a very good efficacy at very less dosage. So then comes this third step, that is medicinal chemistry, where we enhance the activity of your lead molecule. After we have developed a molecule which is very good, which is having all the properties to become a drug and is acting on your target, then we take this lead molecule, this modified molecule, and test its activity in vitro and in vivo. So, till these 5 stages is the preclinical stage. So, after preclinical studies, then we take the molecule, the final molecule which has passed all these levels to the clinical trials. And believe me, after doing so much of research, after studying number of molecules, when we take them to the clinical trial, most of the drugs actually fail in clinical trial. So, this drug development is a very cumbersome field, and sometimes 1 in millions of molecule could actually reach the market and could actually reach the patients. So, now we will talk about target selection in neurodegenerative diseases. Okay. So far you have seen so many different mechanisms. Alan is mostly talking about mitochondria, phagy. So if we see, there are many, because we do not know the main reason how it initiates. So we have to pick something in between, we have to pick anything in between which we think could be a step which is at the very beginning of the disease progression as our target. So when we see all these diseases, let us see Alzheimer's, Parkinson's, ALS, Huntington disease, every neurodegenerative disease have their own hallmarker protein. Like in Parkinson disease, it is alpha synuclein, which is a hallmark protein, and it aggregates, form Lewy bodies and all that. In Alzheimer's disease, it is amyloid beta, tau protein also. They are the reason, they are the hallmark proteins, they are the reason for the progress of these neurodegenerative disorder. ALS is superoxide dismutase 1, this is a copper zinc SOD, and then TDP43. Then in Huntington disease, there is a protein Huntington. So all these neurodegenerative disease, they have their own hallmarked proteins which causes the disease. And if we see what happened to these hallmark proteins, so one thing is common in them, that is they all aggregate, alpha synuclein, it aggregates, it first forms from its native form. When it is in native form, it is important for the system, it is important for neurons, but somehow whatever switch happens, we do not know, but it gets unfolded and then it is misfolded, and because of that change in its folding, it starts aggregating. It aggregates first of all in the form of monomers, then protofibrils, and ultimately it becomes fibrils. Now what are fibrils? Okay, I will talk about it in next slides. So this aggregation is common in all of them. Amyloid beta aggregates in the same form, monomers, protofibrils, and then become fibrils, alpha synuclein, fibrils, everything, Huntington, fibrils. So their aggregation process is same. They all convert their conformation and then become fibrils. Fibrils are the insoluble form of these proteins. Now this could be a target, but there can be other targets also. But let us now, you have already gone through this, the process of Alzheimer's and Parkinson disease. Alan has shown you many different pathways of these diseases. I will just quickly revise it. In Alzheimer's disease, we have APP, amyloid precursor protein, which is a transmembrane protein in the, it is present in the membrane. So this particular protein is in its native form is okay, it does not cause any toxicity, and this protein has some cleavage sites. So if it is cleaved at alpha and gamma site, then whatever product it form is again okay, it does not impact anything, it is non-toxic, and the products are they are soluble and they stay in the cell, they does not cause any harm. But if it is cleaved at alpha, gamma and beta site, so by the enzyme BACE, that enzyme, so then it produces this A beta peptide, a small peptide. Now this small peptide, this has an ability to, this has a property of aggregating. This A beta peptide, it aggregates, it first its monomers from oligomers, then it further form protofibrils and finally it becomes fibril. Then it is dangerous. So now in Parkinson disease, your alpha synuclein protein, it's physiologically, it's a very normal protein, it is very important protein for the synaptic transmission. It is present in the synaptic vesicles, and when it is present in the synaptic vesicles, its C terminal is slightly exposed outside of the synaptic vesicles. I hope you know what are synaptic vesicles. They carry the neurotransmitters which fuses with the presynaptic membrane and then take the neurotransmitter out from the neurons. So the vesicles have alpha synuclein. Physiologically the alpha synuclein is present something like this, but this alpha synuclein actually help in the accumulation of synaptic vesicles, and it helps in the release of neurotransmitters from this into the synaptic cleft. Alright, physiologically it is a very important protein, but again somehow something happens, its structure changes. Then what happens when its structure changes? It get partially membrane bound. Its helical portion is now more exposed outside the vesicle, and thus it prevents all the other vesicles to accumulate together. Alright, and then what happens? And then this alpha synuclein protein, its helical structure shifts, converts into beta structure, and then it start forming oligomers, then it start aggregating. It aggregates into, first it aggregates as oligomers, then protofibrils, and ultimately fibril structure, something similar to amyloid fibrils, which is an insoluble protein. And when it is doing all this thing, it thus damages the neurons. So similarly all other diseases in Huntington disease also and in ALS also, superoxide dismutase, this is an enzyme which is practically present in all the cells of the body, but somehow some switch happens and then this protein also changes its conformation and it starts aggregating. So it looks like that aggregation seems to be a initial step, but again Alan posed a question to you, what do you think, this aggregation is the consequence, it is the reason, or it is both? So what answer did you give? Both. So we actually don't know, we don't know. It is a reason, this is a step which seems like it is an initiator, but once it initiates, it promotes further alpha synuclein's aggregation. So alpha synuclein, once it start aggregating, it gives signal to the cell to make more aggregated alpha synuclein. Amyloid beta once form, it start aggregating, it gives signal to the cell to form more aggregated alpha amyloid beta. So it is both, it is a consequence and it is a reason also to promote its aggregation. So what happens, how this aggregation proceeds? Mostly when the protein is in its physiological conformation, it exists in monomer form and it is normal, it does not do any harm, it is actually doing its physiological role, because see, nature what it do is, nature removes anything which is not required by the body. Slowly through evolution, something which is dangerous, which is not required by the body is removed, but we still have amyloid beta in our body. Why is it there? It has a physiological role. Even amyloid beta is also, and in small amount, it is involved in synaptic plasticity, it is involved. So everything has its role, and when they are in their physiological concentration, when they are performing their physiological role, then they exist as monomers. So somehow something happens and these monomers are mostly alpha helical in structure, and then these alpha helical structure, their conformation changes and they actually convert into beta sheets. I hope you understand a protein exists in alpha helical secondary structure and then the beta plated sheet. So some switch happens and the alpha helical structure of the protein, it gets converted into beta plated sheet. Now once the misfolding, we call it misfolding, of the normal physiological protein happened and the protein is now in a beta plated sheet structure, then the aggregation starts. So the first step of aggregation is one monomer will aggregate with another monomer and will make a dimer. then dimer, another monomer, monomer will make an oligomer. So till oligomer stage it is soluble, the aggregation is soluble. After that then the structure start making a very you can say you know collagen fibers, collagen fiber ka structure, one collagen then binds to another collagen and then they make such a strong protein fiber which is very strong, and it actually if you see it's you apply force on it, you can't break a collagen fiber. Same way these proteins, when they convert into protofibril structure, they start joining with each other and then they make such a strong fibril structure which is like a very strong fiber that they are no more soluble, and then they actually, you know, they cannot stay in the cell, they are thrown out of the cell and they exist in the brain in extracellular space. So these fibril structure, if you see here, so fibril structures are what? Beta plated sheets stacked together, they are beta plated sheets stacked together in a very very coordinated and very, you know, nice what do you call that, conformation, and thus becomes insoluble, and they are very stacked together. So if we see this this particular diagram, so this is actually here we have many different monomers stacked in a fibril form. इसको कैसे देखोगे आप? This is one monomer, this is another, this is another, this is another, and so on. And one monomer is like this. एक monomer ऐसा है, दूसरा monomer उसके नीचे ऐसे stacked है, तीसरा उसके नीचे ऐसे stacked है, and they are stacked like this. All right. So now if we want to develop a drug which could actually, you know, stop the further elongation of these fibrils, then the drug need to attach somewhere on this monomers. So most of the drugs, they are, I mean scientists are working on it, they are developing drug molecules, and if you want to develop a drug molecule which could inhibit fibril formation, then the drugs are made such a way that they bind to the many different monomers stacked together in a fibril conformation. Can you see this? Now, these are different monomers stacked together and the drug binds to them like this. The molecules bind to them like this, and thus it is somehow happening that it is preventing, either it is preventing further fibrillization, or it is binding to it and then it will take it, it will traffic it to some site where it may be degraded, where it may be processed further, whatever is the role of that drug, it will undergo its fate. So now again same thing in a brain, healthy brain, monomers exist in alpha helical structure. While they are in oligomer structure and fibril structures are different. Alright. In fibril structure the monomers are stacked on each other, whereas in oligomer structure their binding is different, and so far we are very unlucky, we do not have the X-ray crystallographic pattern or structure available in the PDB of any oligomer structure of these proteins. But it is found recently, I mean yesterday itself Alan showed you about alpha synuclein oligomer which is present in the membrane. That oligomer of alpha synuclein, it can bind, it can come and, you know, get anchored in the membrane, and it act as a pore through which ions can get in and get out, and that pore is switch on and switch off depending on some reason which is unknown. This is a very latest finding, scientists have found it just now. I mean this paper is published very recently in 2005, in which it is found that alpha synuclein oligomer is more toxic than fibril, and these oligomers, they embed in the membrane and they act as some ion channels, and those ion channels open and close on some signal which is yet to be found. Now the problem is we have X-ray crystallographic data of the fibrils. So far we do not have the structural conformation of oligomers, but it is now found that oligomer stage is much more toxic than the fibril stage. Although fibril stage is also toxic, but the oligomer is much more toxic. So now as oligomer stage convert into protofibril, now you see the stacking here is different, the oligomer stage structure is different and protofibril structure is different. Now once it converts into protofibril structure, then it has initiated a, you can there is a initiator. Now this particular fibril will come start stacking and then it will lead to formation of fibril. So this is the first step of fibril formation. So once a fibril is formed, then this fibril get deposited in the brain and we can easily see the disease in the brain. All right. It's not moving. Okay. So now, now as per if we are trying to find a target of this particular drug, particular neurodegenerative disorder, so far studying all these hallmark proteins. So the first thing that comes to anyone's mind is like protein misfolding seems to be the initiator. So protein misfolding occurs and then the whole progress of disease starts. So this protein misfolding is happening because of which reason we do not know, but these could be the reasons. It could be oxidative stress, it could be genetic mutation, then aging. As we age, since alpha synuclein amyloid beta is naturally present in the physiological brain, but these are the neurodegenerative disease are the disease of old people. So somehow aging is also promoting their aggregation. So then we have a system in the cell which maintains the protein conformation. If that system, Erard system, is dysfunctional, then we will have more misfolding. If there is high inflammation, neuroinflammation, there is any dysfunction in secretory pathway that Alan told you yesterday, that there is a lymphic system in the brain which is actually continuously removing these amyloid beta if they form aggregates, if alpha synuclein form aggregates, its lymphic system is continuously removing it from the brain. But if there is dysfunction in this particular system, then also it leads to accumulation of protein misfolding, and calcium is also very important. Calcium dysregulation also leads to protein misfolding. Now calcium in the brain neurons, it is very important. It is calcium is present at the presynaptic membrane and it is a signal which gives information to the synaptic vesicles to release neurotransmitters. So it is important, but its level has to be maintained. If the cytosolic calcium level increases beyond certain level, then it tends to set the neurons into excitotoxic stage, and when a cell is in excitotoxic stage, then the cells start denaturing. Alright, so calcium is also very important. Now if we see calcium and if we see this Erard mechanism, and then the site where proteins are actually folded, so which particular cell organelle come to your mind? Which organelle maintains, organelle in a cell maintains calcium in the inside it, calcium levels? Endoplasmic reticulum. So which organelle is responsible for protein folding? ER. So considering most of the factors associated with ER, so ER seems to be, again, as we are just trying to find a target which could be a good target for designing drug for treatment of neurodegenerative diseases. So ER seems to be a very good target for developing a drug for endoplasmic reticulum degeneration. So it is found that if misfolded proteins or aggregation of protein is loaded in a neuron or in any cell, it impacts its endoplasmic reticulum and puts its endoplasmic reticulum in a stress. So that is known as ER stress. So once endoplasmic reticulum is in stress, then it starts a cascade of events which is called unfolded protein response, and then that unfolded protein response further takes some information to the nucleus, to different sites of the cells, and ultimately if it is continuing in the cell, it leads to neuronal damage, neurodegeneration, because the cell ultimately goes into apoptosis and thus it degenerates. So protein folding, misfolding impacts ER stress. ER stress starts unfolded protein response, and unfolded protein response ultimately lead to neuronal cell death, axonal degeneration, synaptic dysfunction. But when it is beyond one threshold level, actually again any mechanism in the body, any system in the body is actually made for protection. Why do we have unfolded protein response if it leads to neuronal cell death? It is better to remove such a system from the neurons, but actually it is important for the neurons. If there is some minor stress in the neuron, due to some reason, because it is a machinery can go haywire any time. If there are some proteins which could not be properly folded and they are misfolded in the neurons, in the endoplasmic reticulum, then unfolded protein response actually tends to correct it. It tends to correct it, because what happens, this unfolded protein response gives signal to the neuron to produce more chaperones, to produce something to actually help in proper protein folding. But this till some level. If the stress is so high that it is beyond the threshold, then this unfolded protein response give the signal to the nucleus. Now it is beyond our control, just better destroy this cell, and then it takes the cell into apoptosis. So everything is there for a reason, and adaptive unfolded protein response is actually important for neuronal plasticity. I hope you know what is plasticity, but if it is beyond that threshold level, it actually tends to set the neuron towards apoptosis and neuronal damage. Now what is this unfolded protein response? Now when this is a normal endoplasmic reticulum, when it is working properly, the proteins are fine, they are folding normally. So what we have is, we have these transmembrane proteins in the endoplasmic reticulum. This is ATF6, IRE6, PERK. So these are some transmembrane proteins in the endoplasmic reticulum, and they are all bind to, you can see one particular protein on them, which is, here it is mentioned BIP, it is also called GRP78. So this particular protein, it blocks this ATF6, IRE1 and PERK whenever the endoplasmic reticulum is in normal functioning. So the proteins are being formed and they are properly folded, there is no problem in the endoplasmic reticulum. So these particular things are actually blocked by GRP78 or BIP. So as soon as the proteins start misfolding, the protein loses its conformation and they got converted into some other structure. So then the endoplasmic reticulum undergoes a stress condition, and in that stress condition, this BIP, it leaves these transmembrane protein and goes and binds to the misfolded proteins. Now actually BIP or GRP78 is actually, it's like a chaperone protein. You know what is a chaperone protein? So chaperone proteins, their main role is to help in proper folding of the proteins. It helps to bring the misfolded, it have to correct the misfolded proteins into its actual structure. So GRP78 is one of it's like a chaperone protein, and it binds to the misfolded protein and try to bring them in their proper structure. Either it takes them to their proper chaperone, or if it's beyond control, then it takes it to the Erard mechanism to destroy the misfolded protein. So when the BIP proteins, they leave these transmembrane proteins and goes into the ER lumen, so then what happen, these transmembrane protein, they are no more blocked by GRP78, and now they start their work, they initiate the unfolded protein response. Now what happens in unfolded protein response, all these transmembrane molecule, they further, they go into cytosol, they impact some molecules into cytosol and ultimately cause their impact by influencing the nucleus, and thus whatever was required by the cell that time. If it is an adaptive unfolded protein response, then the cell will start producing more chaperones so that better folding of proteins are made, and if it is beyond control, then it will set the cell into apoptosis. So what happens in unfolded protein response? ATF6, if we look at the ATF6 molecule, so ATF6, when it was blocked with GRP78, it stayed in the transmembrane. As soon as GRP78 left the ATF6 molecule, ATF6 leaves endoplasmic reticulum membrane and it translocates directly into the Golgi. And as soon as it translocates into the Golgi, here the Golgi, it trims it into a active form, and that active form of ATF6 straightway goes into the nucleus, and there it influences the expression of enzymes, expression of mRNA, which codes for more chaperones, and those chaperones then go into the endoplasmic reticulum and further help in proper folding of the misfolded proteins. Alright. So this was all a protective thing. So PERK, on the other hand, when it is bound with GRP78, it stays as a monomer. As soon as GRP78 leaves it, it dimerizes. PERK is actually a kinase protein. Kinase proteins, they phosphorylate. So this kinase protein, it phosphorylates this EIF2 alpha, this particular molecule, it phosphorylates it, and when it gets phosphorylated, this thing further activates ATF4. Now this ATF4, it goes into the nucleus, it do many things. Of course, it helps in increasing the chaperones expression, but it also impacts the apoptosis signaling in the cell. On the other hand, IRE1, this is the most dangerous of all. What it do is, its main role is to cleave RNA. So first of all, it just cleaves XBP1, it's just an mRNA, and it breaks this mRNA into two fractions. So what its main role is? In adaptive unfolded response, it is just meant to break all the mRNAs, so that it wants to stop more formation of proteins, because now endoplasmic reticulum is in stress, it is not able to properly fold the proteins. So this IRE1 wants to stop all the mRNA to go into endoplasmic reticulum, so that it wants to stop the protein formation, it wants to stop the translation. Alright. So it cleaves the mRNA. But sometimes in adaptive unfolded response it is okay, but as it increases, it puts the whole cell, because it is going to cleave mRNA unconditionally, all the mRNAs will be cleaved, so it impacts the overall health of the cell, and thus this after this it activates, it's not mentioned here, it activates another important signal, that is CHOP, which puts the cell into apoptosis. It initiates the apoptosis in the cell. Alright. So this is unfolded protein response. Now so far what you have read is that aggregation is one reason for this disease progress. Now aggregation of these proteins actually put the endoplasmic reticulum in the cell, or endoplasmic reticulum cell puts these proteins into aggregation mode, and whenever endoplasmic reticulum is in stress, it initiates UPR, and UPR also have this particular mechanisms, these many you can see pathways here. So now just keep them in mind, all the molecules you are going through, they can, and anything can be a good target of a drug, but you have to identify which. So this is about IRE1, as I told you. Yes, it cleaves XBP1 and thus helps in cell survival, but this has another way of it. When it cleaves ASK1 mRNA, it actually put the cell, it initiates the apoptosis or cell death, it initiates, put the cell into this particular, put it in on this path. So if we say this, okay, so again under terminal and unfolded protein response, ultimately all these three, IRE1, PERK2, all of them finally leads the formation of small vesicles which ultimately become the autophagosomes. So, as Ellen is telling you about that, under these neurodegenerative disorders, mitophagy happen, phagy happens, and ultimately the cells undergo autophagy. So, if we see the mitophagy, phagy, autophagy, if we go upstream of it, then it seems like endoplasmic reticulum and unfolded protein response seems to be an initiator of these mitophagies and phagy. But again it's hanky panky. It's always like sometimes the answer is it's egg or the hen first, because sometimes it looks like the phagy is initiating autophagy, but sometimes it's like mitophagy initiates or it gives signal to the endoplasmic reticulum to make more such vesicles and bring more molecules into mitophagy and phagy. Now if you see like the thing that I was telling you, now if you see all the diseases, different neurodegenerative diseases, and if we see the hallmarks of those diseases and their impact on endoplasmic reticulum, so there are many studies, and if we try to sum up the studies in one such particular diagram, so we can see that if you see here, it is found that if there is alpha synuclein load in the neurons, or there is Huntington load in the neurons, or VAP-B load in the neurons, it influences endoplasmic reticulum signaling, and it influences unfolded protein response. It actually enhances it and thus give the signal to the nucleus to start the unfolded protein response expression of mRNA. Alpha synuclein Huntington also influence the export of proteins from vesicle into the Golgi. It is found that amyloid beta, prion and Huntington, it impacts the calcium hemostasis. So far role of alpha synuclein in calcium hemostasis is not found, but amyloid beta can impact the calcium hemostasis, and majorly it increases the calcium load in the neurons. Same way it is found that Huntington and tau protein, not amyloid beta, tau and SOD1, it influences the Erard mechanism. Erard mechanism, now what is this? This is again a quality control check of endoplasmic reticulum. If there is a load of many misfolded proteins in the endoplasmic reticulum and the chaperones are not good enough to bring them back to their actual structure, so those misfolded proteins, they are sent to the Erard mechanism, where those misfolded proteins are destroyed and removed from the cell. So this is a quality control check of endoplasmic reticulum. So these proteins, Huntington, tau and SOD, it impacts, it slows, it reduces the activity of Erard mechanism in endoplasmic reticulum. And it is also found like alpha synuclein aggregation, when it starts, SOD1, prion, and this aggregation once it starts, it impacts the folding of the proteins in the endoplasmic reticulum. So again the same thing, whether it is the folding, misfolding that initiated alpha synuclein aggregation or alpha synuclein aggregation which initiated the folding, they both are interconnected. Alright. So it is found that once we add aggregated alpha synuclein to the neurons, it turns the switch on in the endoplasmic reticulum to make more unfolded or misfolded proteins in the endoplasmic reticulum. Alright. So these are the things that happens in the endoplasmic reticulum under different disease conditions when their hallmark proteins are exposed to them. Now till now, what do you think could be the best target for therapeutics of neurodegeneration disorders in ER? Now just it's some point for you to ponder. What do you think it could be? If we somehow manage the Erard mechanism, can it be a good drug target for this particular disease? Yes. Which is the best? Splicing. Okay, splicing, for splicing what we have to do? It is the IRE1. So what we have to do is we have to inhibit IRE1.
I'm saying IIM because it has a major role and it is also highly conserved. So I think increase its chaperone activity, maybe
I mean, if we want to, you know, make IRE1 as a drug target, so what we are going to do is we are going to block it so that it should not be able to splice any mRNA. So will that be really good target for this drug, I mean for this disease management? What do you think? What others think? Just ponder what you have, I mean you have, I have told you some mechanisms in it. So now what could be the best way to treat it? Okay, what if we block it?
But if looking at Iran mechanism, so that is essentially degradation, which is in our favor, which I understand. But if this happened with previous mechanisms that are going wrong, so that the protein is being misfolded, so that raw machinery is going to continue making that misfolded protein, and even if we are just discarding it, all we are doing is wasting resources.
Absolutely,
So
very good.
either the folding part is addressed, because discarding it again and again, it might help जब disease हो चुकी है, but not good.
So you are saying that we should not put more effort on Erard mechanism because we should, this is something which is coming after the protein is misfolding. So we should look something here, that something which should stop protein misfolding. Okay. So what do you think should be the target? Think. There is no wrong answer. There is no wrong answer. Anything can be a protein, I mean drug target. Chaperones, okay. So these chaperones, okay. If we can alter, what do you want? We should increase their activity?
In that case, that GRP78, which is getting detached from that membrane in the proteins. So if we can inhibit that process.
Okay. If we increase the amount of GRP78, and then it will block the unfolded protein response. Okay. So now what will happen? You have misfolded proteins, alright. You have some misfolded proteins, not much, and you have increased GRP78 which will block ATF4, PERG and IRE1. Now what will happen? The misfolded proteins are like that only, they will stay in the endoplasmic as it is, and you have stopped the adaptive UPR. Adaptive UPR was actually good. It would have actually worked by enhancing Erard mechanism, by enhancing other chaperones, and by enhancing, I mean other mechanism to maintain protein folding structure. So what we did is we just stopped one step. I mean there is no wrong answer, but I will tell you what, everything has some drawback. The point is this, every strategy has a drawback. That is why we are not able to find a drug so far. So this way, but the misfolded protein load is still there in the endoplasmic reticulum. We just stopped UPR, but some UPR was also required.
Ma'am, one of the safe approach to do it will be like managing the calcium homeostasis, because all other mechanisms have some intrinsic role, they have physiological role. And if you are going to turn off the switch permanently, then in case we actually need some unfolded protein response for some other toxic proteins, etc., then it would be harmful in that way. Also, now the relationship is pretty bidirectional. We actually don't know seeding is leading to this, or either this is leading to seeding.
Yes.
So,
Okay, yes.
responsible in calcium dissociation, like AMPA, NMDA, and several other iron receptors. So if you could manage some of the receptors, we could target the calcium dissociation to some extent.
Okay, so you are saying, you said many things, but I'll just pick one thing. You are saying if we could stop the calcium influx in the cell. So now what about, what is the role of calcium influx? why we need calcium in a neuron? As I said, it is important. Yes.
Targeting the dyshomeostasis, not like
Okay, how could we maintain hemostasis? Very good. How could we actually maintain hemostasis?
by, for
We can't block the channels because channels need to work. We need appropriate amount of calcium in the neuron. We just can't stop it. But we need to maintain its hemostasis. Its level must be maintained. How could it be maintained?
there is some inherent mechanism, for example, inhibiting the TRPs channel inside the cell. Or actually, it's quite interesting because if we inhibit amyloid beta or tau or whatever protein, it also indirectly inhibits the calcium because
So,
there are multiple studies.
So, yes. So now you yourself gave an answer. We cannot just inhibit the channels because they are important. They are actually maintaining the calcium hemostasis. We are, this hemostasis is actually lost because of the protein misfolding. So now you gave a target, that is amyloid beta aggregation and or alpha synuclein aggregation. So can we find anything here as a good target? This is the mechanism. This is the mechanism, and everything is actually important. We can't stop one thing for the other and other. But yes, although I may be wrong, but so far, Hanji.
Can we increase the receptor like sensors like IRE, like instead of lesser of IRE1, if you increase the receptor like IRE1 protein. So in this one, like you're not even stopping anything, but you're increasing this mechanism. Like I'm thinking in case of more transcription factors and chaperones would be useful.
So you are saying increase the expression of IRE1 only or chaperones also?
Only for IRE1.
Only for IRE1.
You're saying UPR is not able to press, like it has a threshold. So I'm saying if you increase the IRE, so the threshold might increase. Like you increase this threshold point. Let's say for
No, if we increase IRE1, what will happen? We will destroy more mRNAs, and but destroying mRNAs which are required for vital functioning of the cell is also not good, isn't it? So we have to actually limit it. We don't want to destroy all the mRNAs in the cell because there are many other vital functions like glycolysis, Krebs cycle. These things must be, these things must continue. Their enzymes must be continuously synthesized. So we should have some, translation also occurring in the cell, even though the cell is in stress, I think. It's good. It's good. Just, we should think. It's very good. I'm very happy that you are thinking on some terms. Give a hypothesis and then try to find its limitation yourself. Okay, if we'll do this, then what could happen? Oh, these things can happen. If we'll do this, then what could happen? These things can happen. Give your hypothesis and find its limitations. This is how your brain should work. When you are studying any disease, when you are studying its mechanism, you should see every step, every pathway's metabolite as a drug target, and then see that if I target it, what benefit it could give, what drawbacks it could bring with it. This way we can actually narrow down to what is best for the treatment of the disease. All right. So far everything is so complicated, and anything can be a target, but anything can, they have their own limitations.
We can also use microRNAs here, which can suppress these proteins.
MicroRNAs to suppress protein aggregation.
So that
Why you want such an expensive thing? Okay. Okay. I thought you were going to say microRNAs to suppress gene expressions. There are treatments, there are studies going on in which microRNAs are being explored to manipulate gene expressions. So again, that becomes a genetic therapy, translating it is a challenge, but yes, there are many papers you will find in which microRNAs are also being explored for the treatment of neurodegenerative disorders. So, it's okay, I think you have thought I have given enough food for your brain. We can move forward. Now, let us talk about the chaperones in the endoplasmic reticulum. So, now there are many resident chaperones in the endoplasmic reticulum, and their role is to give proper folding to the protein. One of them comes under a class of heat shock proteins. So, the heat shock proteins like heat shock protein 70, it helps in folding of the protein, and it is dependent on ATP molecule. When it is bound to an ATP molecule, it is in an open form, and then the protein comes and get attached to it, and then ATP is removed, then it tightly binds the protein. Whatever changes in the protein structure happens there, and then ATP is again regenerated, and then the folded protein comes out of the HP70 chaperone. So, sometimes HP70 does not, cannot fully give structure to all the proteins. So, partially folded protein then goes into HSP90, and HSP90 is very important. It gives mostly, it ends up all the protein in their proper folding structure. HSP90 is actually a dimer, and the protein gets attached in the cleft between these two dimers. ATP molecule is consumed to give the protein its proper structure, and finally a proper folding of protein is achieved. Alright. So, there are other chaperones like calnexin and calreticulin. So, these chaperones are basically involved in glycoprotein structure. So, they are mainly involved in adding the carbohydrate group on the protein structure. So, if you see most of the these transmembrane proteins, they all have glycoprotein, they all have a carbohydrate group attached to them. So, calnexin and calreticulin also have an important function. So, this is just a basic idea about the chaperones, and these chaperones are natively present in an endoplasmic reticulum, and they are doing their work. Whenever the misfolded protein burden increases, their expression increases to maintain the misfolded protein burden. Okay. So, there are the molecular chaperones are the chaperones which are natively present inside the endoplasmic reticulum, and how they work is due to any stress condition when the proteins are unfolded or misfolded, these molecular chaperones, they come and bind to it, and then they form a protein complex with it, and then they turn them into their functional state. If these chaperones are less in number, or unfolded proteins are more and chaperones are less, then these proteins, these unfolded proteins, they start aggregating, and thus they lead to this particular disease condition. Now, apart from molecular chaperone, there are other class of chaperones, that is chemical chaperone and pharmacological chaperones. They are not normal resident chaperones in the cell. They are drug molecules, chemical molecules, which have ability to do the same thing as the molecular chaperones do, and they thus become a very good candidate for the treatment of this disease, to actually block the aggregation of these molecules. So, if I see all the mechanism, not I, many other scientists, if they see this mechanism, so they found that if we could somehow reduce the aggregation of these proteins, then we can treat this particular disease, and chaperones become a very good method of treating these aggregations. Now, we cannot make the molecular chaperone as targets, because molecular chaperones are again proteins and very big proteins. We can't use protein as a drug. We need its alternative, and we have many alternatives so far. Since last many decades, scientists are working on it. So, we have chemical chaperones, we have pharmacological chaperones, and these molecules, they actually mimic, they work in the same way as the molecular chaperones. So now molecular chaperone is clear to you. What are chemical chaperones? They are some chemical moieties. They are not specific, they just don't bind one particular protein and give it its structure. They act same as the molecular chaperone. They help in giving the structure, protein folding structure, and help in its proper folding non-specifically. Then we have pharmacological chaperones. So these chaperones are some molecules which are specifically designed to fold specific proteins, and there are many reports in the literature where natural compounds from like ginkgo biloba, curcumin, so these compounds are also found to have ability for enhancing protein folding, and they seems to be quite specific, as these extract like TCM, it binds specific to some particular proteins. So if you see the mechanism of action of these chaperones, it's clear. The chaperones, what they do, they directly bind to the misfolded proteins, and they also reduces the endoplasmic reticulum stress, and thus impact the UPR and reduce the burden of aggregated proteins, and thus treat the disease. So once endoplasmic reticulum is maintained, it also will reduce the burden of, it maintains the calcium hemostasis. So these are certain, there are many chemical chaperones known so far. These are the most popular one: TMAO, trehalose, TUDCA and PBA. So these are some molecules. Now you see their structures, they are not so complex. So these molecules, they actually act as chaperones, and they are very helpful in maintaining proper protein folding. So if we see their mechanism of action, so I'll skip this part. So they are, I told you, there are some of them are specific, some of them are non-specific. I'll straightway come here. Now trehalose and glycine. So these they are osmolytes. Osmolytes directly attach with the misfolded protein and help in its proper formation. Alright. Osmolytes has just one role, has just one mechanism. This is mentioned here only. So I'll just show you in this slide. So they have just one mechanism, that is giving the protein its proper structure. So they don't have any other role. So osmolytes are very good in giving protein structure when we use them in vitro. But as soon as we use osmolytes as a drug in the cells or in the animals, it's very difficult to translate them, because they don't have, they are not specific, they don't reach to the target site, they don't have any other mechanism by which they can help in the treatment, and an osmolyte, one osmolyte molecule should actually reach the misfolded protein to cause its action. So it's very difficult to translate it, but osmolytes, when you put it with an unfolded protein, it helps in its proper folding and it prevents its aggregation. So apart from that, these two molecules, PBA and TUDCA, they are found to be very good chaperone molecules. So TUDCA and PBA, there are certain reports which says that they directly bind to the unfolded protein also and prevents its aggregation, but they have multiple roles. They are also involved in reducing the inflammation. They are also involved in, they goes into the nucleus, like TUDCA, it binds to this ferronoid X and does impact the expression of certain genes in the nucleus. And your PBA, it binds to HDAC, histone deacetylase, and it impacts the expression of some genes in the nucleus. So they have multiple roles. So PBA, not only that, PBA have another role. What PBA do, if we have unfolded proteins in the endoplasmic reticulum, they are not encapsulated in the vesicles. Only the properly folded protein molecules get encapsulated in the vesicles and are removed from the cell. But when PBA is present, it encapsulates the unfolded proteins also and removes it from the endoplasmic reticulum, thus reduce the load of misfolded protein and endoplasmic reticulum. So PBA has multiple roles. So, considering all these properties, now if we see the molecular structure of TUDC, it is very complex, and this seems to be simple and it has many other properties. So, we can zero down on the molecules, So, in our lab we zero down on 4-PBA. Now, 4-PBA is a molecule which is a very small molecule, it is a short chain unsaturated fatty acid. This is actually an FDA approved drug. It is used for uric acid disorders, not for neurodegenerative disorders, but for uric acid disorders. It has a very high LD50, so it is not toxic at all, and there are many studies which says that it has good chaperone activity. So, we studied its activity in our lab also. This is my PhD student who studied the role of PBA in excitotoxicity. We induce excitotoxicity by giving them AMPA, and what we found was that PBA actually it reduces endoplasmic reticulum stress in excitotoxic animal models. It maintained calcium influx of endoplasmic reticulum stress, and it also improved the cognitive abilities of the animals in excitotoxicity. So, it seems to be a very good lead target. Now, if we consider aggregation as a target and we want to prevent aggregation of molecules, and then PBA becomes a very good lead molecule. So, if it has such a good property, why it is not yet translated as a drug? So, this is the outcome of that study. I will just skip it because we are short of time. So, now why we could not translate it? So, the reason is, although PBA has a very good activity, it is very effective, but the dose which caused this efficacy was around 200 mg per kg weight in animals, and if we translate this drug to humans, then it will come out to be around 3 grams per day. So, a drug at this high dosage, it is not feasible to be ingested by a patient and to be used by a patient. So, its dosage is high. So, the first and foremost step which we need to do is, we need to develop a molecule. Now, if you see my first slide, we have selected a target. The target is aggregation. Now, we found a lead molecule which actually stops an aggregation, that is PBA. Now, we will improve it. Now, PBA needs improvement because its dosage is very high. We need to make it in such a way that it shows the same efficacy with less dosage. So, how to do that? We have to make some new molecules, we have to make new derivatives of PBA and then test them. So, how would you do it? You will start randomly making derivatives? No, you should have some basis. Yes?
Take some molecules that does not produce any other side effects, like bioequivalent. So, modify the molecule as such that it has no side effects.
Okay, how? This is your PBA, how will you modify it? This.
So, ma'am, by analyzing the structure activity relationship, then keeping the groups same which are important for their management, and
Yes.
deriving a small.
Yes, we need to have a base, we need to have some information that we can't, if we randomly change it, yes we can, when we do not have any information, we have to randomly change this molecule, we have to randomly add many different groups, but if we have some idea, then it helps your study. So, there is a study by Memori in 2012. So, he gave a very good, good outcome of his studies. He said that we can enhance the activity, anti-aggregation activity of PBA if we increase the aliphatic chain in PBA. If we add a methoxy group at the para position in the PBA, it enhances its anti-aggregation activity, and if we add more electron clouds, benzene rings to this molecule, it enhances its anti-aggregation activity. So, these findings were given by Memori, and on the basis of these findings now we can modify this protein, this molecule, and we can add chains and keep on increasing. We can add benzene group, we can add methoxy group, and so far we can change. Now we know what changes we need to bring in this molecule to enhance its activity. So what we did, some of our students sitting here in my laboratory, they made a huge library of this modifications. They made a library of around 30-40,000 molecules, and then they tested its ability first of all in silico. So how this is done? So now we can't do these changes in each molecule and then test it directly on the animals or on the proteins. It will be very time consuming and it will be very uneconomical. So to bring it to make it more economical, we use these in silico tools. So what these in silico tools are, we design a huge library in silico. So after lunch we will have a session of this in silico drug designing. How many of you have experience of in silico drug designing? No one. So this next session will be really helpful to you. So what we do is we make a huge library of molecules. We should know what changes we can actually do it. If we don't know, then also it's okay. We randomly change the molecule. So after that we test the binding efficacy of those molecules with the target protein, how strongly that molecule binds with the target protein, and then then those molecules which showed very good binding with the target protein, they are tested for their ability to become a drug. Like sometimes, like if you see this TUDCA molecule, this is a very good molecule, but it is very complex. It cannot actually be translated as a drug. Why? Because it will have it has low blood brain barrier. It cannot cross the blood brain barrier. So if our drug cannot go into the brain, then it's of no use. So we need to make such molecules which has good absorption. They should be easily absorbed in the intestine. They should have low toxicity. They should be able to cross the blood brain barrier, and they should have other best properties to actually become a drug, and that also is studied in silico using ADME analysis. So you will study that today after lunch. We will have a session on that also. So once we screen all the library, we see which molecules best bind with your structure, then we see which molecules has best property to become a drug. They can cross the blood brain barrier, they have less toxicity, they are less absorption, they have more absorption. So those molecules are then selected and synthesized in the lab. Alright. So when we synthesize these molecules in the lab, normal chemistry reactions are done to synthesize them chemically in the lab, then their structure is validated by spectroscopic methods. Here I am just showing FTIR, but we do NMR, MS, other methods also. So the structure is validated and then they are tested in vitro and in vivo. As I have shown you in the first slide, we select the target first, then we select the lead molecule. Lead molecule is PBA. It has some problems, we need to improve it. We improved it, we are now improving it. We improved it through in silico studies. We synthesize these molecules now, and now we will test them on in vitro platform. In vitro platform in our case will be what? We are testing them on, we are testing their ability to impact aggregation. So our in vitro platform is anti-aggregation ability. So now what we will see is how well the molecules are binding with your target protein in vitro directly, and are they actually preventing aggregation. So for that we will straight away take either amyloid beta molecule peptide or alpha synuclein proteins and expose them with your drug molecule, induce aggregation in them and see whether your molecule is actually inhibiting the aggregation or not in vitro. And there are other methods also, instead, like we are making a non-specific chaperone molecules. So, it is not only impacting alpha synuclein or amyloid beta. So, we can also study them on any other protein. We can take BSA, induce aggregation in them and test our molecule whether it is inhibiting the aggregation in them or not. So, this is in vitro studies, and after that we take it to the animal. In animals we induce the disease. We can either chemically induce the disease. I will tell you about this animal models and animal studies in my next talk to you, on which is on Friday. So, I will just give you a brief idea. We induce the studies either chemically, like for Parkinson disease we give rotenone to the animals, this induces Parkinson disease in them, or we can take transgenic animals. Although now transgenic animals are considered better model for neurodegenerative diseases. So, then we check our drug molecules whether they are actually helping in reducing the this disease load in them by various parameters. And after we complete all these steps, so we then only then can narrow down on a molecule which has crossed the preclinical 5 steps. We have a molecule which could reduce this disease burden in animals. That molecule can go further in the clinical trials. All right. So, that is it. Thank you. Any questions you have? Yeah.
As you said, that you are checking the binding of the molecule to various peptides. So, for example, amyloid beta has several forms. For example, amyloid beta 25 to 35 peptide, 40 peptide, 40 peptide. So, we have to test individually in every peptide that which one has better binding.
Why should we use, I mean not all peptides are having, you know, aggregation potential. It is 142 is the most prone to aggregation. So, we should make a drug which could actually bind to the most aggregating peptide.
But in the animal forms, we don't use the 40, we use other forms like 25 to 35.
No, we can use 142 also. We stereotactically can inject this 142. We have that model only, and that initiate the aggregation in the animals. In animals we use 142 also. Yeah.
Then, you did the study after your in silico screening. How many molecules did you arrive at?
Okay. Actually, after in silico studies we get, we just put them in order of their best binding ability, and then we check their ADME scores, their ADME properties, and we have to see the feasibility of their chemical synthesis also. So, it all depends, the feasibility and all. Sometimes you can synthesize 15 molecules, you can synthesize 25 molecules, you can synthesize 4 or 5, it varies from the structure to structure. So, in our study I think we have zeroed down to 4 molecules, 4 molecules.
In vitro was done with all the 4 molecules.
All the 4 molecules, yes. In vitro study was done with all the 4 molecules. Any other question?
So, yes, definitely one way is to have all the modifications done in the molecules or find this. Or else, one way to do it also be having combinatorial therapy where 2 molecules or 2 compounds essentially enhance each other's effects, or not each other's, the other one's effects that we provide. So,
Which is, you are asking if we give a combination therapy, is that better, or we should modify the drug molecule? Okay. For combination therapies, this is my opinion, like they should have different targets, like both the drugs should not be acting on the same target. If they act on the same target, then they are competing with each other, isn't it? So, now our target is, when our target is 1, that is aggregation. So, here we we cannot have a combination therapy, but like if we want to target, let us say hypothetically we want to target aggregation and we want to target, let us say, calcium hemostasis, then we can use a combination therapy. But again, you know, for combination therapies, it gives good results. I mean, you can find results sometimes, this is happening, this is actually it's giving a synergistic outcome. But if we want to see the mechanism, sometimes some mechanisms are interlinked, sometimes some mechanisms are not interlinked. So it's sometimes very difficult to pinpoint that what could actually have happened. But there is, I mean, research are going on, people are working on it. But when you are trying to find a new molecule, when you are trying to find a new drug, I think at that time you should focus on one target. So once you are confirmed that your one target drug is best, then you should try different combinations with it. And when you are doing combination therapy, then your target should be separate. They should always be different. Never use one target as, I mean, never use two drugs acting on the same target.
How about like for this example, we are targeting aggregates. So aggregates are going to be broken down. Is that what we are looking at, or are we looking at stopping the formation of aggregates?
Very good. So this is very important. So Alan has shown you some drugs, monoclonal and antibody based drugs like Wemby. So their mode of action is same. They are going to stop the aggregation of these proteins. Like Wemby is going to stop aggregation of amyloid beta, and so far we don't have any drug for alpha synuclein. Yeah. So those drugs are actually effective when they are given at the early stage of the disease, because these molecules actually they prevent the aggregation. But once the protein or peptides they have actually come in the fibril form, it's very difficult to break them. It's very difficult to break them. So this therapy can work if we start giving them at an early stage, when these molecules have just started aggregating. Prior to, once a fibril is formed, yeah, once a fibril is formed, it's very difficult to break a fibril. Fibrils are very stable structure. We can actually prevent its further growth, and we can stop the aggregation at the initial step. They are not harmful. That's what limbic system of the brain do. It's cleaning the brain, continuously cleaning the brain.
Many times both of the systems go wrong at a similar time frame.
With the age, this limbic system also gets slow. But once your once you have given a drug which binds to your monomer and is preventing its aggregation, so now that that monomer will not, whatever harm it's going to do to the cell, it won't be able to do that. So what will be its fate then? The cell will recognize it as something foreign and will destroy it. And there is a system in the body to remove the debris. I think that's it. Or anything else? Any further query? No. So I really want you to bring your laptops in the afternoon, but we are having that problem. CamDraw problem is solved or not? Huh? It is solved. So don't leave just now. Shivani will tell you something about installing a software in your systems. You might have brought your laptops today. So after lunch we will have a session on in silico drug designing, where we will just give you a brief idea how could we design drugs and how could we work on it. Alright. So just Shivani एक बार just इनको बता देना, ठीक है? So now we can meet at 2:30 sharp. Please be here by 2:20 and we will start the session at 2:30. All right. इसे रिमूव ऐसे ही कर दूँ बस, ऐसे ही कर दूँ बस। एक बंद कर दो ना, नहीं तो इनको होगा। हाँ, एक मुझे बता के रख दो ना बाई चांस। ओके,
ये ऑफ
डन।
है।
You want me to do the both directional discussion or the single side discussion? बताइए, what do you want me to do? You want me to give an interactive lecture or one side lecture? हाँ, it should be. जवाब ही नहीं आ रहा, पीछे से नहीं आ रहा। मेरी रिकॉर्डिंग हो रही है, जल्दी बताओ। You want to do the interactive session. Interactive is that sometime in between I may ask for the response from you all, right? Right, because without your participation I cannot make it interactive. Is that okay? No? No? Okay. Okay. ठीक है. So very good afternoon to all of you, and as a duty has been given to me that an in silico tutorial for computational approaches in drug discovery, right? Tutorial means definitely it's not the teacher role. Tutorial में हम during our classes in our graduation and in the post graduation term, when we used to run the tutorial, it is by the students, like we flip the classes, right? So you should be on this side and we keep on discussing, we keep on giving the inputs what the student is discussing and all. Now under this umbrella I have given the topic that computational approaches are accelerating the drug discovery research. But before that I would ask you, anyone or who one is knowing the meaning of drug discovery research? Anyone? What do you mean by the drug discovery research, if I ask you? Anyone? Okay, at least one response I expect from the audience. Anyone who can tell me what is a drug? At least one response from the audience. What is a drug? So drug, now let me you accept agree with me or not, all the drugs are chemicals, but all the chemicals are drug. Which statement is true? All the drugs are chemical, but all the chemicals are not drug, right? If I ask you, definitely you must be, the audience sitting over here would be of the different ages ranging from 20 to 25, and in the journey of 2025 you must have come across with any one tablet, a very one common tablet, paracetamol, Disprin is there, painkiller, diclofenac is there, ibuprofen is there, you must be using. So definitely it is a chemical, but how does a drug differ from the chemical that makes it different? So a chemical is called a drug when it interacts with a particular receptor or the target. Is that okay? Right. If any disease is there in the market, if I say that I want to, disease is what? Any abnormality
of the imbalance, any abnormality or the imbalances of the hormones or the biological conditions or the physiological condition is resulting into a condition of called the disease. Now I want to cure that disease. For that disease I said I am commonly using the word that I need a drug, I need a drug. Now what is drug? I need to discover a drug. What is that? I need to discover a drug. Now in our pharmacy there are two process steps, drug discovery and then drug development. Anyone know the difference between this drug discovery and drug development part? So this is a picture. When any disease is in their market, if I being a pharmaceutical chemist, if I being a medicinal chemist, I want to design a drug, I want to provide a cure for a particular disease, I should know what is the disease. You people are biophysics, you people are from pharmacology, you know that what is the difference between the physiological condition and the pathological disease. Any disease is generally like why it is happening. Until unless you don't know the reasons, until unless you don't know why the symptoms are happening, until unless you don't know what is the biological pathway or the target is being involved, you won't be able to design a particular chemical that is possible to go to the site and interact and it give a therapeutic response, then it is called as a drug. Is that okay? So journey starts from identification of the disease. If any one of you in the we people are into drug discovery, I am a medicinal chemist, so my area is to provide a solution for a particular, I am not saying I am the direct person, it is a collective science, right. I will show you one picture where how the journey of the drug starts, from where the journey starts. So the journey starts from if there is any disease in the market. Now try to find what are the reasons, what are the symptoms, what are the possible pathways that involve, what is the target for which I want to design the drug. Then medicinal chemists try to find a suitable chemical that got the tendency to act on the receptor, then it would be tested on the animals, followed by preclinical phases, phase 1, phase 2, phase 3, and I hope you all know that what are these phase 1 and phase 2, phase 3. Depending upon after the successful results in the animal, we put it and give it to the healthy volunteers to take the toxic effect, then we check it on the patients with a smaller number, then we give it to the bigger number in the hospitals. Ultimately we go for we find the investigational new drug form, and ultimately when this part is there, till the molecule is being discovered, chemical, it is called the drug discovery process. But conversion of that chemical into tablet, into the syrup, into the injection or a suitable formulation that it can be given to the patient, that part is called the development process. Is that okay? Fine. Now drug discovery, right? You might have heard about the quinine. You might have heard about the quinine. Quinine is natural or synthetic? Natural. So the thing is, the journey, we chemist people are like randomly, I will come to you what is this terminology is called when all of a sudden you people are starting any new research. But for all of us, very basic research that is being inspired from nature. Okay, so quinine, it was around 1837 when the 400-year back when the cinchona bark was used to treat the malaria. Are you aware of this fact? 400 years back. Then it took 200 years to discover that quinine is present, to isolate the quinine from the cinchona bark, to discover, to establish its structure and to establish its mechanism, and ultimately after 237 years, it took how many years? 237 years. By that time, the person who must have discovered this, he might have died, right? But he has given a blessing to all the humankind. Is that okay? So just note down the journey. So drugs are nothing but, if you look into the market, all the drugs are small molecules, and maximum 99% of the drugs are organic in nature. Okay? Then they are complementary in shape to the target. This is the very important. If I have designed certain molecule and if it is not fitting to the receptor, this chemical will remain a chemical. Important is that a chemical to show a therapeutic response, its complementary shape to the target is very important. Is that okay? I always make it understand in the form of a lock and key. A lock will open until unless there is suitable key to give a noise. Is that fine? Okay. So, oppositely charged. If I say that here is a molecule and here is a target. Now, it's okay, they are complementary in shape. Shape में fit हो रहे हैं. But what about the charge? If they are both of the opposite charge, will they attract each other? No. There would results into the opposite. So, that is the basic fundamental of a drug is that any chemical that got the tendency to interact with the receptor that is complementary in shape and opposite in charge. Now, in your terms, what are the targets? It could be enzymes, you people are working on the enzymes, it could be biomolecule, it could be ion channel, it could be transporter, DNA, RNA, and I think audience sitting over here are exploring the different fields. So, you all are working under the different, someone is working on diabetes, someone is working on the neuropharmacology and different concepts are there. Is that okay? Now, so nature is the one that inspires us, but problem is that if I cut, I hope somebody of you sitting in can imagine that if I cut one plant of kilos, I can get the amount of the chemical quinine in milligram level. Now, in order to serve the nation of 140 crore की हमारी population के अंदर है, what do you think? It this way I would be able to remove the gardens. I need to clear all the medicinal gardens. It would be free of the cinchona. So that is not the solution. So nature inspired me that quinone is present in the cinchona bark. Now in the chemistry lab we need to find out the solution how quinine can be synthesized. And do you know that what is the better version of the quinine that is being synthesized, that was synthesized in the lab? Anyone? Chloroquine, hydroxychloroquine. So these are the two examples that were synthesized by the chemist in the lab, but after the inspiration from the nature. So this is how the journey of the drug goes. Now drug discovery, no doubt, nature is the one of the biggest source, right? But we cannot use the nature, as I told you, the resources are very limited, okay, right? And other approaches is that penicillin by serendipity, right? By chance discovery is there. One microbe say we extracted out the penicillin and that penicillin was used to kill the another microorganism. That is one of the approach. Another is that screening wide variety of the drug obtained. Artemisinin, again artemisinin, I hope you know that it was around 1967 when Dr. Y2 from Japan started working on the 2000 herbs, and then ultimately he reached out to the 640 herbs that show the potential in malaria, and then it was around 2006 when he got the artemisinin isolated, and 2015 he got the Nobel Prize. कितने साल की जर्नी हुई? 50 or 60 years. So 50 to 60 years, like starting from the nature, then find the structure establishment, then mosquito role, mechanism establishment, and hold this to get into the FDA approval, formulation development, and after all ultimately acknowledgement in the term of the Nobel Prize. It took 60 years of the human life. Fine. Chemical modification, मैंने अभी आपको बताया, quinine, chloroquine and hydroxychloroquine, they are the modification. Quinine was a nature inspired. Chloroquine and hydroxychloroquine, they are the synthetic modified derivative of the quinine. Then combination of the two drugs. Sometime, now the malaria is one of the example. You might have learned about ACT, artemisinin combination therapy, right? Malaria, when the quinine was being used, mosquitoes getting resistant or they are getting immune, we are getting like mosquitoes were getting resistant to that, right? So that was an alarming situation to develop new drugs. Artemisinin came into it. Artemisinin is having a short life, but quinine was having a long duration. But in terms of potency, artemisinin was more active, quinine was less potent. So this gave a new thought, okay, let's combine the two drugs. And this is also drug approach, yes, in order to cure any disease. So they thought to let's combine the two drugs that will act in a two different way. So that's why number of the diseases are presently being cured by giving the two medicines rather than one. And last one, where we are turning around, rationally drug designing. Now when we were at your stages, like long 20 years back, when we did our PhD, we used to go to the literature in the chemistry lab department. I remember we used to go to the chemistry lab, and in a one day, single day, we hardly make it possible to check one journal. Like if I am working on the prostate, hardly in a one day, if any, like let us say 2024, 12 volumes are there. I could make it possible to check 12 volumes only. To check prostate is there, prostate is there, prostate is there. Manually we used to do it. But now you people are just a click button away. At that time, we keep on synthesizing the molecules without knowledge, because we were not knowing the receptor structure. So we kept on synthesizing the molecules, let it say 100, thousands, and then in my 5 years, because practically let admit all of us, a PhD is journey of 5 years, right? If any one of you are working on any one target, at least in a 5 years, you can explore one pathway. Is that okay or not? Right? It's not possible, because one particular disease can have a number of pathways also. But in a given period of span, you can manage to study one pathway or one parameter or 2 to 3 parameters. So we used to, at that time, we were not having knowledge about the receptor, we were not having knowledge about the targets. So we kept on synthesizing number of molecules, and my junior will come and he will then synthesize, or I would be submitting my sample to the CDRI Lucknow or to any another institute, pharmacology people, okay, you please test on these molecules, and after 5 or 10 years he will give me some outcomes. So a timeline. Now reverse is happening. After 10 years of spending, I come to know that my molecules are not working on that receptor. So what is happening? 10 years of my life, 5 years of my life, no doubt I have been awarded my degree. But if I talk about the success, how success is being defined? Success is that how the society is being benefited. Okay, no doubt every observation in the research makes a big contribution. There is a journal of negative observation also. Please always never drop your idea, because if I have worked, I have spent my 5 years of research on those molecules, it means I am passing my information to the coming generation, don't work on these molecules. If I would not be doing the publication of the negative results, people will start again doing the research on that part. Okay, so the rational drug design is that I would be starting rationally, logically, after knowing the structure of the target, what is the protein structure, and how this is how now we are stepping into it. Now look into this picture. I would ask you at the last, how many molecules we have synthesized? 10,000 or 1 lakh molecule. After 1 lakh molecule, hardly 1 or 2 molecules make it path to reach to the market. 1 lakh molecules are in the lab. I am talking about in the lab, and it's not possible for me to synthesize one molecule, 1 lakh molecule. But researcher like me, who all are working in the area of the prostate, they must be working in the different area in the prostate, but different skeletons. Now I have combined the information from all these molecules. Let it say this data has been collected, and do you know what this data is called as? If I am collecting the chemical information from all of you, let it say you all are medicinal chemists sitting over here, and you all are working on 10 different 20 skeletons. You all are synthesizing different molecules. Now I am keeping this pool at one place. What is this base? What is called? Yes. What is this called as? Now it's a database. Database. Kiska? Chemical ka. So chemical database. Same way you people have the database also. Kiska? Biological database. Don't you have? There is a target proteins. बहुत सारा है. Database क्या है? Right? It's a collection of that. Na? Google is one of the biggest database these days. Perplexity is there. Chatbot. Number of the resources are being there. Now look into the success rate. After like 3, 6, 5, 10, 1, 2. overall it's a 18 years of minimum 18 years of journey is required for a chemical to become a drug after spending $1 billion of the dollars on it, right? And you cannot deny that you all are using the animals for bioassays. Maximum. We medicinal chemists may or may not use the animals, right? We can skip that part because our part is to synthesize molecules. But those molecules hold value until unless they are not being tested on some animals, they are not having any therapeutic value. In order to check therapeutic value, definitely I am using the animals. You people are using in order to study the in vitro, in vivo, to check the therapeutic efficacy, in order to study the whether they are physiological, in order to study the what is the fate of the drug in my body, then bioassay, preclinical toxicity, for any kind of like studies you people are using this. And yes, same way you cannot deny that somewhere we are torturing the animals. We are torturing. I am not saying, we also did it and we have to do it to some extent, but is there any way that we can curtail it on it? Yes. So in the laboratory they are deprived, sometimes we make them deprived of the food, sometimes we are giving some injections to induce the disease. This way it means the animal is under pain, under distress, and in order to perform, like I have said that we need a skilled person. I am a medicinal chemist, I am not the right person to do the animal study. I would be in the need of the help from you people. So there are certain issues while handling the animals. I am not saying that we can, neither computers can replace the humans nor the computers can replace the animal studies. But there is a way. We need to find the alternative. I hope you have heard about this 3R strategy. R means reduction, refinement and replacement. How we can reduce the number of animals? How we can modify the biological method that it gives the less pain to the animals? Is there any alternative that I can use and I get the same observation of the results? And the answer is lying in the. So I hope you are in vitro models, you know that. In vivo models, you know that. Now the biological evaluation using the computers is called the in silico. Here the two branches joined hand together. What type of two branches? Computer sciences and the chemistry people, or the computer department and the biological sciences. Without these two, computer science, biological, stats, math, algorithms, everything is being required. I am just giving my data to the computer peoples, nothing that, beyond that. Computer people does not know about much about the benzene structure even now. For them benzene is what? 01010101. But how that benzene is being drawn, that is different for them. But I can see only 6-member ring with 3 double bonds. It all depends on the perspective how he is looking and how I am looking at that person. Okay. So it is the integration of the computer people and integration of the biology or the chemical or the stats or the physics. All the integration of all the branches is being required. Right. Now I am giving my data, I am collecting all, let me say all of you people are collecting your data, you are working. Now you people are providing this data to the IT people. IT people are storing that data as a databases. Now again the next generation will come, it will put some keyword into it, it will try to retrieve the information and try to utilize this knowledge into your field. So this is how implementation of the integrated approach, that is called the in silico tools, data is being stored, information is being extracted out, and you will apply your knowledge. Don't leave everything on the machines, don't leave everything. Okay. So you need human integration is always being involved, always. If you will say ki let do the machine do it, PhD degree he will get it. Okay. So human intervention is always being required. Computer sciences, chemical sciences, biological sciences, physics, stats, math, all are working together, all are working together, right. So this is how integration of this approach will help you to minimize the mistakes, to make the faster decisions, and in order it will reduce the time to reach to the market. I will show you how it become. Initially I show you the slide where the 1 lakh molecules were being synthesized, and hardly after 18 years 2 molecules were reaching to the market. But after the computational from past many years, since it has driven into the market, around 45, after 45 molecules, 23 molecules are showing success, and out of 23, 5 molecules have already gone into the market. So can you compare the 1 lakh molecule versus 45, 2 molecule versus 23 and 5 molecules? Success rate, if you look into the percentage, it is the 5 molecules over 45, that was 1 over 1 lakh, right. This is how the computers look into. But आप लोग क्या देखते हैं? बटरफ्लाई. आपको क्या दिखाई दे रहा है? आपको 01 दिखाई दे रहा है या फर्स्ट टाइम में क्या दिखा रहा है? वेरी फर्स्ट टाइम. इल्यूजन इमेजेस आती है ना, इंस्टाग्राम पे आती हैं, what your eye sees was. So we being from the science people will try to have a look on the mosquito rather on the 01. I'm least interested in how the 0101 is being displayed. But what is catchy to me, to my eyes? It is the bacteria. So this is how benzene structure, chemical structures, like it is growing field, algorithms, mathematical instructions are required. Researchers have integrated techniques, artificial neuronal network, formal logics, mathematical stats, operation research, economics. You cannot say that. You cannot skip the psychology, physiology, linguistic, neuroscience. All fields are working together. All fields. If you really want to get the best use of it, so it is the one of the biggest tool. So computers increasing, computers are not restricted into the, like you people are using on your mobiles, you are using on the Instagram, Facebook, like weather forecast estimation is there, number of that booking hai. You people are using the technology, or you would like to say you people are living the technology? It's a better statement that you people are living the technology. So technology, like it has gone in, it has delved into the number of the applications of the computer in the healthcare. You know that disease identification, CT scan is there, it's computer. Doctors are keeping the records, it's computer, right? Smart health records, medical imaging, then drug discovery, manufacturing, disease predictions, everything. I hope you are aware of the, like China recently 10 days back has introduced the AI machine in the market. You just stand in front of that machine, it will diagnose your body and it will give you the blood reports and all the tests that what kind of disease you are having. Are you aware of this technology? 10 days back. And I hope also you know that China has introduced the hospital with 40 robots where no single doctor is there. So what are robots? What is that machine? Ultimately it is IT, and IT is what? Computers. You can use the word AI or you can use the word computers, but working it. But the thing is, how that machine is working, how it is able to, if you look at, you stand in front of the machine and it will help you to diagnose the, like somewhere data has been driven, data has been added. Based upon those algorithms, it will try to predict your disease. Algorithms तो लगेंगे ही लगेंगे, data अंदर जाएगा ही जाएगा. Data is important, integration of all the branches is important. If I say that, open your mobile now, right, and just Google out single word paracetamol. How many pages you would get? Hurry up, anyone. Kitne? Just I can tell you, right? It will give you approximately 1,20,000 pages. Honestly admit that how many of you have explored second page? No. we go look at the first pages and 20 pages, अगर 20 sheets आती हैं पहली sheets पे तो हम 20 को ही देखते हैं. It's very true. हाँ, filter लगा लीजिए, आप फिर भी चले जाइए. Still you don't have that stamina to go and look into the 1,20,000 pages, right? Now that paracetamol word is there, right? Your interest is of the paracetamol. Now from where this 1,20,000 pages are coming? I am chemistry person, I would be looking into the synthetic approaches. You people are pharmacology people, you will try to look into the like how it is acting, right? Doctor is looking ki what kind of the latest information side effects are there. So everyone is working on it. Biochemistry people will try to look at another way. The same 20 pages are there, but everyone will try to look from its own angle, right? Fine. अब उसी को देखने का when after finishing this class, I will show you one more dimension how to look into that page. Okay, so this is like how the computers are entering and keeping a hold on our daily lives. So computational tools in the drug discovery process, it has become need evolution is not a process force, ज़रूरी नहीं है, but it has become the part of the journey, right? We are not saying to use it, but over a period of time you need to apply it, because FDA guideline has now been approved to use the computer predictive data. I will show you that slides also. Slowly I will come. So high, because this use of this computational tool will high attrition rate. Attrition क्या होता है? Failure or success rate? Increased expenditure. It is being required to control, to address the issues of high failure rate. Attrition means failure rate. I was synthesizing 1 lakh molecule. Now after use of the computer, if I am synthesizing only 45 molecules, don't you think my expenditure, there will be a curtail on my expenditure? Yes, there will be curtail. Increased volume of data, definitely volume मेरा data बढ़ जाएगा. Increased globally requirement, ठीक है? Requirements अभी recently, if you might have seen that one of the syrup has been banned by the FDA. Cough syrup is there, ठीक है? So the moment any side effect is there, FDA come into picture and it immediately banned. उन्होंने पूरी पंजाब में बैन कर दिया है उस सिरप का यूज़ करना. Why? यूज़ करें तो रेगुलेटरी रिक्वायरमेंट्स जो होती हैं वो टाइम टू टाइम दे कीप ऑन चेंजिंग, राइट? सो चैलेंज इन द टाइम, द बिगेस्ट इज द टाइम प्रोसेस. If COVID is over here, if I would say that I need to discover a drug, and in order to discover a drug I am in need of the 3 to 4 years or the 10 years, by that time world would have been swept out. Agree or not? Anyone interest, anyone can tell me COVID की वैक्सीन को इंट्रोड्यूस करने के लिए कितना टाइम दिया था? Yes? No idea? क्यों? ड्रग रेगुलेटरी तो कहती है कि मिनिमम आप 15 to 16 years clinical trial. So that was the regulatory decision on immediate pandemic basis. Otherwise they are not allowed, supposed to release the drug in the market, right? And how that drug was, how they have discovered the drug that can control the COVID? Again, that was the computers. Okay, because you people are from different field, I will try to make it interesting by showing you the one example that's true from the COVID, जिससे हम सब effect हुए थे accordingly. Okay. So now here is the drug development. Earlier I showed you this slide where the traditional conventional waves were there. Now look into disease से लेके clinical trial तक, all the phases have been taken up by the computers. What are these terminologies? Bioinformatics. Informatics, what do you mean by informatics? Bioinformatics, it is the integration of the IT and the biological data. Cheminformatics, integration of the chemical and the IT. Reverse docking, protein structure, target, library docking, de novo, pharmaco, QSAR, structure based, in silico ADME, physiological. So these are the all computational tools and the approaches that are being in trend to fasten up. If you look, remember my first slide, computational tools are accelerating drug research. Accelerate क्या होता है? Fasten up. they are helping me to fasten up. So all these tools are, it's not like you can use maximum about 2 to 3 approaches you can apply your during PhD projects, right? And sometime if you look into the, from today onwards, maybe the vision you get changed in silico, in vitro, in vivo correlation, like what you have predicted from computation, try to correlate with the biological data and then try to establish a relation. So this is how it is called the in silico in vitro correlation. And sometime medicinal chemists use it in a vice versa, designing, synthesis and evaluation. In our time we used to say it was synthesis and evaluation. We used to keep on synthesizing molecule and we keep on evaluating her. But now the concept is designing, synthesis and evaluation. Now what is the designing? I am designing the molecules hypothetically on the computer. Then I would be testing it on the computers. Those molecules that will be showing the good results on the computer, I will pick it into the lab, from dry lab to wet lab. Journey is called dry lab to wet lab. In silico, in vitro correlations there. So you might be, you people have seen number of paper, but शायद हम सुनने के बाद वो और comfortable हो जाता है. Okay. So these are the various tools, and it is would be very tough to cover all these tools, but let me try to give on the structure based drug designing. This is the one of the tool that you all may need over here. Structure based drug designing. Now structure based drug designing is the tool. किसकी structure? Whose structure? If I ask you, very good, it is the structure of the receptor, right? If now what, according to you, what you all are biological people with a background, what do you mean by the receptor target? What is that?
Protein.
It's a protein. And if I have further break down the protein, what is protein? It's amino. If I further break down the amino acid, what is that? No, my dear. Look into the chemistry part. It is a carbon. It's having one H, then amino group, carboxylic group is there, and one R is there. So one amino acid differ from the another depending upon what R is there and how they are the uski stereochemistry kaise hoti hai. Is that okay? So that is the amino acid. Now your genomic structure, how it differ from each other? आप लोग बहुत ज़्यादा बायोलॉजी के पर्सन हो, हाँ बताइए. How my features do you differs from you? How? Genes, यही है ना. Genes तो वही हैं, basic तो 4 bases तो वही हैं. Then still how we differ, 20 के 30 people and we don't matches with each other. It's a rearrangement, right? Now if you work on any target, you pick any target that comes to your mind, जो भी आप जिस फील्ड पे आप काम कर रहे हैं, उसकी कितने अमाइनो एसिड्स हैं? Anyone can guess? Rough idea? किसी का रिसेप्टर हो? नहीं पता? कितने? 51. Now do you remember the sequence? फिर? So how it will, don't you think it is important? Now you people know that protein is like you people draw the structure in a one dimensional way. What is one dimensional way? Amino acid, amino. Now 2 dimensional way, then 3D, and in your body it is present in a 2D, 3D, 4D, what? 4D. Right? Don't you think the folding of the protein and the sequence of that amino acid makes a difference? So it means if I know the structure, if I know the sequence of the amino acid, how they being are folded within my body, will it help you to design your molecules? Will it help you to design your molecules? Now what is this called? This is called kiski structure. So that is called the structure based drug designing approach. You know the defining the binding geometry. If you remember my first slide, I told you that drugs are nothing but the small organic molecules that are complementary in shape to the receptor and that would have the opposite charge. So it means if my amino acid is having NH group like that and the carboxylic group is on the back end side, so it means while designing a molecule, don't you think if I am having prior in hand information, what kind of the groups are present over here, can I introduce that kind of the groups that immediately? So what I am doing? I am defining the binding geometry. So this is called the structure based designing. And here you people, this is one of the biggest people for the field for the biophysics people and the biochemistry people. You people are required here. You know that the chemistry, I am not technically sound in this case of this light. You know that the protein structure establish करने में आप लोग, I think you people are playing a big role in that. You know the chemistry, उसकी resolution चाहिए आपकी, right? Sequence is important, resolution is important, NMR data is important, right? IR, X-ray crystallography is important. So you people are doing that. You people are developing the machines. You people are establishing this high resolution requirement is that. Now 50, he said कि like ma'am insulin is having 50 amino acid, but he doesn't remember the amino acid sequence, but computer does. Not only computer does remember 50 amino acids, but he can remember like 256, let us say if any receptor I am working, it is working on the 250. You people have come across with the different isoforms of any enzyme. You know that? There are isoforms of the enzyme, right? What is the difference in the isoform? How you define the isoforms? COX-1 and COX-2. You might have heard about it. Anyone? What do you mean by the isoforms of the enzymes? How they differ in the different? Yes? Same gene number, उनका amino acid sequence same रहेगा. उनकी working condition, उनकी presence. Like let us say if I am working on one enzyme, it is present in my body, in male human body, it is present at the 3 different stages. One is present in the prostate, one is present in the skin, and one is present in the hairs. So don't you think if I would be designing a molecule, if I design a molecule for the prostate, there is the information regarding the enzyme present in the prostate or on the skin or in the hair matters? Will it increase the specificity or sensitivity? It will increase the specificity. I am designing the molecule specifically for the prostate. So it means these are isoforms. Is that okay? So this is called. So pocket topology. Now how this isoform, the isoform present in the prostate is present in a different conformation, that present in the hair and that present in the, so that is called the pocket topology. Okay. The molecule that can interact with the receptor present in the prostate may or may not be, should not be able to interact with the, it should or should not? Should not be able to interact with the receptor. For that what is important? Pocket topology. For that what is important? High resolution, conformational state. So well defined pockets are there or the disordered pockets are there. So computers are continuously helping you to predict, to give you the data on daily basis. Earlier times mein you people are, this is, I am really telling you that number of jobs are waiting for you in this area. Go to the LinkedIn and looked in for the jobs for the SBDD or the computational tools or the chem informatics people. You people can help and enter and take the over there. LinkedIn पे जाइए and number of jobs are waiting for you. But only thing is you need to just do the some small AI. कोई भी 1-2 महीने का course कर लीजिए, you can do it. Okay. So well defined pockets and yet the information is being given. Another is ligand based drug designing. Okay, I will come it on a later end because it may not that help you again. So you might have heard the word docking. So structure based drug designing is nothing but the docking. Structure based drug designing is nothing but the docking, right? Simple ways, I will always try to make it very simple way, lock and key. If you are having 20 keys and 1 lock, by looking into the keys, can you make me understand कि किस type की key लगेगी lock को? I hope you can. क्यों? How come? If you are having a bunch of 20 keys and there is a 1 lock, can you filter out 2 or 3 keys based upon just looking at the lock and the key concept? Yeah. By size, right? By a roughly idea about the pocket, by looking. So now I will take back to you, the lock is here your enzyme, that hole on the lock is called the pocket, and the keys are the chemicals. Is that fine? So now this docking is a paradigm shift in the drug discovery. It is being, it's not simply merely like, it is being used by many ways, polypharmacology, drug repositioning, virtual screening, targeting phishing. Let it say if you have one molecule, but number of the, let it say 230 receptors are present there, right, databases में से, this is called bioinformatics, databases, PDB databases or the biological databases or the protein databases. If you have 230 proteins are there in the database, but you have one molecule, आपका बहुत प्यार है उस molecule के लिए, you said I want to do something for this molecule, right? Now what you will do, you will try to, this is called the reverse docking, you will try to check the interaction of this molecule towards all the 230 proteins. And with the best interaction, you can say this particular molecule got the potential to work particular against particular type of disease. Are you getting my point? Is that okay? So that is the target phishing or the profiling or virtual reverse docking भी हम उसको बोल देते हैं. Prediction of the adverse drug reaction. Now if he says that insulin, I said कि ma'am I want to do the like let it say any I said कि insulin के लिए काम करना चाहते हैं. He knows that my target is for insulin का जो भी इनका receptor है, right? So molecule should interact with that particular receptor, right? But I am checking, I am putting a model while evaluating or while doing the PhD. I have tested my molecule only for that target. How you can be sure it is not working for another targets? बताओ. मैंने अभी बोला कि PhD में प्रैक्टिकलिटी रहती है, 5 years की प्रैक्टिकलिटी, कि PhD करनी है 5 साल है, लेकिन उस 5 साल के अंदर आपके पास एक पाथवे होता है, right? That was the biggest question to me during my PhD viva. I always share this, it's an inspiration for everyone. He said, I said कि my molecules are very potent for the prostate cancer. He said कि okay, how come? I said sir, I have tested them for the prostate. So he said, did you test it for other cancers also? मैंने कहा no. So then it means in order to ensure that whether it got the potential to work on the other sites also, it means I need to check on all those also. Now this can be, it means I need to check, this is called the polypharmacology. Polypharmacology. Not exactly polypharmacology, I may present it. Now this particular molecule, if it is showing, let it say I have checked it for the lung cancer, colon cancer, skin cancer and the prostate cancer. If it has shown very good activity against only the practically in the prostate cancer, it means it's really good. If it is showing some of the activity against other cancers also, then it means it got the tendency to work on the other targets also, right? Now effect on other targets may be desirable or non-desirable. If it is non-desirable, then it means you are checking the drug interact with toxicity effects. Undesirable effect is called toxic effect. Let it say if I want to do this study in practical on humans, on animals, it may take how many years? Whatever the period I have shown you. But all this you can do it by using simple computers in 1 month. So adverse drug reactions, polypharmacology, he said I want, let it say you said ki, I said ki 5 alpha reductase or the enzyme is present in 3 forms. I am in my 5-year tenure, it is possible for me to check only one pathway. But my examiner, my reviewers are asking, have you checked it for the another 2? It is not possible practically for me to do all this study. Definitely next junior student will come and do that. But by giving the predictive data, doing the studies on the computer, I can give and defend myself that yes, I have done the computational studies, predictive studies are there, my molecules hold the potential, and that's why we can do for it. Is that? So polypharmacology means if your molecule got the tendency to act on the number of the targets rather than one. Now the action on these additional target may be desirable or undesirable. If it is undesirable, then adverse drug reaction. you may do the target fishing. Fishing में क्या होती है? Receptor. Fishing क्या होती है? You pull the hand, right, and pick out one fish. So it means if you are having one hook, that hook is your molecule, and amongst the 230 receptor, your receptor has shown the activity against the one. It means you start your journey with this molecule against that particular receptor for a particular disease. So this is our virtual screening. I will show you one examples. Drug repositing. Repositing मतलब क्या होता है? Any idea? Yes, Shabash.
Using it for another
Yes. And any example for this? Very good. Very good. किस चीज़ के लिए? Very nice, very nice. Yes. So like drug is already. Do you think this will help in saving the time and the resources and the toxicity data? Aspirin was there earlier, it was for the headache and whatever it is. Now it is being used as a for the blood के लिए use करते हैं. So whatever the data was studied on the patient, on the animals, on the clinical trial, the same data will because it has been studied for those. So repurposing is one of the again another attraction of the science and the research these days. Drug repurposing. अगर FDA site पे जाते हैं, FDA की site पे जाइए and you will look into that number of the 40 to 50% of the drugs अगर clinical trials में पहुँच गई हैं that are based upon the repurposing. Again by using the tool computational. Which tool? SVDD. SVDD means docking. Is that? So if you want me to explain, these are the various steps before doing that we need to prepare the protein, we need to prepare the ligand. It's very important to find out the binding site. Then we need to remove our water or what is the importance of the water or not, right? Then molecular docking and ultimately score is being done in the form of the like G score and so on. So these are the basic steps. Better it would be if I will be showing you one example from my lab, and also if I ask you, here different two type of populations are there, or you can say three. One is male and female. Another is age factor के हिसाब से. I am in the age of 50, you people are at the age of 25, right? And there are geriatric, pediatric and adult and the young ages are there. If I want to send, if I want to sell a drug, if I want to bring a drug into the market, don't you think I need to study the drug for all these age factors? I need to study how the drug will behave in my body, right? Biochemistry people, I have synthesized, let it say 50 molecules, and on one molecule one student can do the PhD because he will be doing the study how it is going into the body, different compartments, blood, urine, serum, like what is happening, what are the different metabolites, what is the journey, half life क्या होती है, कहाँ कहाँ travel कर रहा है. So there are the number of the way. So even one molecule के ऊपर I have synthesized 50 molecules in 5 years. Now from one molecule one PhD students can be trained, because it's not only the PhD trained, but the thing is, in order to bring that 50 molecules into the market, I have to collect the data based upon their absorption, distribution, metabolism, protein binding, you know that, right? So if the drug is getting metabolite, you know that. If I am having, you might have heard about that, someone doctor says that take this medicine with food, take this medicine without food. What is the reason? So don't you think again we need to look into this factor that I am giving the, again I need to do the studies in the practically life way, right? So if I want to give this, like give the drug to empty stomach or stomach, right? It means food is affecting somewhere absorption, okay? This is one of the parameter. Now if a healthy volunteer is there and one is diseased person is there, right? Dose is different for a childhood, dose is different for geriatric, and dose is different for the adulthood. What is happening in the geriatric people? They are getting old, metabolism getting slow down. Small baby is there, metabolism system has not developed yet. We are at the perfect age. So don't you think before, again once I will quote that, before bringing the molecule into the target, I have to provide all this data? Yes. Thousands and thousands of the papers are required in order to bring one molecule, and number of years and billion of dollars are there. Now with the help of computers, if you look into chemical spacing information, so एयरलाइन के लेफ्ट हैंड साइड पे इज द इनपुट डाटा एंड द लेफ्ट हैंड राइट हैंड साइड machine modeling is there that will help you to predict. This is called the ML and AL. You might have learned about all these things. You might have come across these terms. These are AL, है ना, algorithms, and the machine AI and the ML. So chemical specific information. Whatever the scientists so far have developed different molecules, they are predicting the molecules or the data in the lab, and they are doing the physicochemical properties, partitioning behavior, tissue permeability. I am giving my all this data to the IT people, right? Plus I am adding the information about male, female, age, organ, kidney, liver, blood distribution. So this is physiological and anatomical feature. I am giving the power to the computers that this is the data and this is the chemical space information and this is the physiological. Now computer will do the algorithm and the calculation and it will predict a model. Now you bring any molecule from your end and you ask the computer that based upon this model, please what help me in getting the information, what would be the possible future of this drug in my body. And in order to do this study, I need minimum of 5 to 7 years. But a click away and I will try to get it done now. हम इसके बाद हमारी एक छोटी सी एक्सरसाइज प्लान की है जहाँ पे हम ये वाला काम करने वाले हैं. We will be trying to predict the properties of the molecule. That would be my part. अभी हम जो ट्यूटोरियल में करेंगे, you will be doing that. So in silico I have provided. अब आप कहते हो मैम सॉफ्टवेयर है. सॉफ्टवेयर में क्या है? सॉफ्टवेयर में गार्बेज इन गार्बेज आउट, जो इन्फॉर्मेशन आप डालोगे बेस्ड अपॉन दैट मॉडल ही वो डेवलप करेगा. यू माइट हैव वर्कड ऑन सम मॉडल्स, किसी ने तो डेवलप किए होंगे ना मॉडल. मॉडल कहाँ से डेवलप होंगे? अंटिल अनलेस आई एम गिविंग द इंफॉर्मेशन और द डाटा टू द कंप्यूटर, फ्रॉम दैट इंफॉर्मेशन ही इट कंप्यूटर विल ट्राई टू डेवलप द मॉडल बेस्ड अपॉन दैट प्रिडिक्शन. आई विल मेक आउट द प्रिडिक्शंस. I will meet up. So chemical information, secondary drug screening. Very first screening is that, मैंने आपको पहला बताया था, very first screening is that this is docking is the very first. These days if you look into the any papers or into the docking is being used as a primary drug screening approach. Second screening process these days being implemented is that ADME properties. Okay, this is the oral. So I want to show that. Let us say if you people are doing the in vitro or in vivo enzymatic assays in your lab. I am working with Dr. Tanzee, just like I have synthesized molecule. For me, my research is done because for 5 years I am struggling to synthesize one molecule. But from that one molecule, ma'am did so many of studies, like I am not in position to recall those pathways, sorry, I won't be able to do that, right? I am not the right person to comment on. So if you look into permeability, then transportation, melanin binding, ultra equilibrium dialysis, you people are doing PCR something, etc. So all there are tools, computational tools for everything are there, everything. Only thing is that you just Google out and ask for the AI or the computational tool for your study. I guarantee 101% you will get the tool for that. Some are free, some are paid. The difference between the free and the paid is that free में data updated नहीं होता. Let it say if she wants to do the ADME study by using the free software. क्या फ़ायदा फ्री सॉफ्टवेयर का? बिकॉज दैट इज़ नॉट द अपडेटेड वन. ओके, उसको इतना बेनिफिट ना. बट द कमर्शियल जो होते हैं, पेड होते हैं, उनके अंदर लास्ट मिनट तक का डाटा. सो इफ शी विल बी प्रिडिक्टिंग द इंफॉर्मेशन, if she is doing the research for PhD or really for the industry cannot afford the free softwares. Industry is paying लाखों करोड़ों रुपए पे कर रहे हैं for their softwares. Okay, so one molecule against ADME की वजह से in phase 37% molecules get fails in the ADME. So in our body on an average 40 organs are there. If I want to say that, okay, you do the insulin, during insulin you are studying one organ, कौन सा, वो भी नहीं करना, नहीं कर रहे ना. So it means in order to bring your research into practically into the market, so I need to, there are 40 organs present in our body and 400 cell types are present in my body, 400 types of cell, and FDA needs data on each and every organ, each and every type of cell before it comes into, before I can file the IND. 400 cells को study करना, right? Not at all feasible for one person, not at all possible for one institute also. Collectively we are working in different directions. But so there were, these days computational tools are there and they are helping you to do this. Another tool is that homology modeling. In case you are not one, we have also used these tools, homology modeling. Have you heard this word? Have you heard this word, homology modeling? I say that SBDD. What is SBDD? Structure based drug. Kiski structure? If the structure of your receptor is not known and you want to develop what would be the possible structure of your target, then the tool is called the homology modeling. Homology means same. Model means I am developing a model. So homology model will help you to develop the 3D protein structure of your receptor in case you PDB. What is the full form of PDB? Protein database. If the protein, do you have any idea how many यहाँ पे मैंने शायद लिखा होगा यहाँ पे yes, over 200 million sequence known versus only 22 lakh solved structures. Million कितनी structures हैं हमें जो solve करनी है? How many structures? 200 million sequences are yet to be resolved, and how many have resolved so far? Only 2 lakh. Okay, yet so much of work is pending and you all can contribute into this, right? So homology content, X-ray, crystallo, NA, cryo, these are costly techniques, no doubt. In order to establish the structure of the protein, you need to isolate, you need to crystallize it, you need to establish its NMR data, and all this can be done by using the computational tool, and that is the 3D structure, that is the homology modeling. Dr. Tanzi, please let me know the timing. Shall I? 5 minutes. So I will be showing one case study from the lab, so they get the interested. Okay. So let me take you to first. Yes. कोविड इंडिया में हमारे पीयू में कब लगा था लॉकडाउन? 17 मार्च. आई रिमेंबर डॉक्टर तंजीर और हमारा शुडेंजर लैंड किया था. वी पेड 6 to 7 lakhs दैट टाइम. द मोमेंट इट लैंडेड इधर से लॉकडाउन हो गया. डॉक्टर रिमल इज विटनेसिंग. 7 lakh we paid for this software, right? So I was getting continuously call from Schrodinger's head ki ma'am please do some studies for the India. China has already published 500 papers. 17 मार्च तक, बाय द टाइम जब तक हमें ये समझ आता है कि कोविड हो गया है, लॉकडाउन करना है, वी नीड टू क्लैप इट डाउन, वी नीड टू कैंडल डाउन द लैंड. दैट टाइम, बाय दैट टाइम चाइना ने 500 पेपर्स पब्लिश कर दिए थे बाय यूजिंग द computational tools, and it was the computer power only that they established the structure of the coronavirus too. It was the only structure. I think यहाँ पे मैंने लिखा होगा जनवरी 2020. जैन में 2020 में कंप्यूटर्स के साथ. Now what is this? This is the, if I want to bring the structure, if I want to design, COVID की वजह से number of deaths were happening, I want to have a control on the number of the deaths. What I need? I need a drug. मुझे stop gap arrangement चाहिए था. अब before that कि वायरस तो होल्ड में आ ही नहीं रहा था, right? Virus, it was not possible to hold. जैसे वायरस आ रहा था वो पर्सन किल हो रहे थे. So the moment they said कि okay, let coronavirus SARS-CoV-1 भी है आपको, if you remember, based upon the knowledge of the SARS-CoV-1, they predicted the possible structure of the SARS-CoV-2 and they use the structure of this SARS-CoV-2 and they, please look into the figure. Look into the figure. 1.3 billion compounds. 1 पॉइंट किस टाइम तक ये पेपर आ गया था? 11 मार्च को. हम 17 मार्च को अंदर गए थे. 11 मार्च तक 1.3 बिलियन रैपिड आइडेंटिफिकेशन ऑफ द रेमडेसिविर का नाम सुना है? कहाँ से आया था? यहाँ से आया था. After screening 1.3 billion compounds after the possible predicted structure of the SARS-CoV-2, right, remdesivir was one of the, यहाँ पे I think ये रहा, hydroxychloroquine, PGI में hydroxychloroquine and remdesivir. I remember my one of the colleague was the very first COVID patient in Chandigarh, Dr. Gurpal was there and he was being given the hydroxychloroquine and that time it was given. So that is the success of the, that is the power of the computer. अगर हम वेट करते कि कौन सी ड्रग दे, नो डाउट रेमडेसिविर स्लोली इफ यू रिमेंबर इट स्टार्टेड, ब्लैकमेल भी होना शुरू हो गया था. 25,000 की पुणे की एक सन की स्टोरी भी है जिसमें सन ने कहा कि प्लीज़ मुझे दे दो एंड आई फील सम टाइम बैड, इट इज़ आवर जनरेशन ओनली और या हमारी ही सोसाइटी के अंदर डॉक्टर हैं. ही सेड आई विल गिव यू एट द कॉस्ट ऑफ 2 लाख. लाइक rather than saving, it was a point of business for them. So remdesivir and hydroxychloroquine timeline shows 3 COVID pathway and this was for the infographic. This was the outcome of the, and if I ask you the question, how the remdesivir was discovered, not discovered, like identified. यहाँ पे discovery और identification में फ़र्क है. Please note it down. I have not discovered. We have not discovered. What we have done? We have identified. From how many compounds? 1.3 billion compounds. वो compounds कहाँ से आए? From the chemical databases. And anyone knows the biggest chemical database? Yes? Biggest chemical database of free zinc database. If anyone wants to explore, number of molecules are lying there. Just category wise, skeleton wise, receptor wise, pathway wise, try to explore the zinc database. Zinc database. ठीक है, I would be giving the, so remdesivir was identified based upon the knowledge of the target. So that's why it is called the target based drug designing. I hope whatever the information I have tried to share, yes, these are some of the pictures. Computer models are replacing the animal testing. We know the facts. FDA has already given the guidelines. Please give the predictive toxicology data rather than putting the animals or the humans on the stress or unwanted pains you are giving that. Please give the predictive data. All those rather than you testing the 1 lakh molecule on the humans or the animals, please give the short list out the 40 molecules from that. So they says that FDA is saying that, if I have circled it, I hope you can see it out in silico. I hope you can find it out, right? So FDA has already, what is FDA? I hope you remember, you know that it is one of the biggest, right? So FDA is already given the instruction, please start giving the in silico data, means use the computer power and give me the predictive data. Don't spoil our resources and try to save the human lives. ठीक है जी, and no doubt software built are continuously validated and we refined these are in silico toxicology. You can look into the last corner, pharmacology. In silico toxicology means you are predicting the toxicology of the given chemical by using the computers. Slowly and slowly softwares are getting refined day by day. Yes, cost and the data, robustness and the algorithms will vary from one software to another software. Let me admit that. If I am using one software free, what is the paid? कहाँ 7 lakh, कहाँ free है? Don't you think the reliability of the data will vary? Yes, it will vary. But for the beginners, we always recommend to go for the free softwares. Once you get comfortable with that, shift it to the paid one. If you are really doing the research, that is going to make a big difference in the society. Okay? Certain facts and challenges while using that, अभी हमें पता ही नहीं थे, right? A lack of knowledge and awareness of the suitable AI tools or the हमें अभी नहीं पता. In fact, if I say कि even I am fully having knowledge, no, sorry, only 1% knowledge I am carrying. So whatever knowledge I am carrying, I am just trying to transfer it now. You people are at the young stage. A lack of trust, yes, a lack of trust is there. Somebody guide नहीं कहते नहीं करना, is there, है ना? A lack of trust towards the AI or the bias towards in vitro and in vivo आता है. Overemphasis on flashy pilots at the expense of easily scalable solutions. ठीक है? Overemphasis on the flashy pilots at the expense की like I have to do this, है ना? Inadequate technical infrastructure. गवर्नमेंट फैसिलिटी है पूरी पीयू के अंदर हमारे पास 2-3 सॉफ्टवेयर्स होंगे, हमारे पास कितना होता है? प्राइवेट सेक्टर्स में जाइए, दे आर हैविंग दिस मच ऑफ स्क्वायर. फ्री ऑनलाइन वर्सेस पेड, आई हैव ऑलरेडी शेयर दैट. सॉफ्टवेयर हैज़ इट्स ओन शॉर्टकमिंग्स एंड पिटफॉल्स. डेफिनेशन ऑफ द एंड यूज़र, मैं कैसे देखती हूँ, आप कैसे देखते हो, राइट, डिपेंडिंग अपॉन द नॉलेज अप टू विच एक यंग जनरेशन को दूंगी और एक स्केलेबल है. Then softwares in silicon model might not perform well if a predicted chemical is beyond the chemical. Yes, this is one of the, like I have Dr. Rimal is sitting over here, one of my student, and I have case study from our lab also. In our case, he is working on the neurodegenerative diseases like the target, like what is the aim of your GIAN school. So neurodegenerative, he did what? He designed a library of 10,000 molecules on the Schrödinger, right? Now he did the docking, okay. He did the ADME, okay. And ultimately he filtered out somewhat 25 molecules. But we cannot ignore that what computer is giving, whether it is actually feasible in my labs also. Sometimes flashy, not flashy in this way, flashy in terms of computers also it can happen. So we need to that it is sometime beyond the chemical space. So we need to filter out, no doubt out of 10,000 he filtered out 25 molecules, and amongst 25 molecules he filtered out, we may left around 3 to 4 molecules, best molecules, because that complexity of the reaction was not feasible in our reaction. So ethical and the legal issues, whenever we people are using, आज की डेट में तो you people are using the ChatGPT, you people are using the Perplexity and many more softwares are there. Earlier इसको AI को क्या माना जाता था? Illegal, right? And I hope you know that now the guidelines, the journals have allowed to use this tool in case if you are quoting them as a reference. You can use them. You can use them. There is a limit. Percentage of the limit is there in the journal that you can use these data, ChatGPT, Perplexity or whatever the grammar what you are using. But there is a limit and the cap on the uses of these, and when you are quoting and giving the reference that I have used this from ChatGPT, you are safe. You are safe. Is that okay? So, repurposing. Shall I take 2 minutes more? Can I take 2 more? Huh? Now here is a one example in silico identification. मैंने क्या word use किया है? Identification and experimental validation, and I am giving the exercise name as repurposing. This is practically we have done in our lab. Okay? So what we did it, you people are like Parkinson? Huh? Oh, no, I am just talking about this disease, Parkinson. So, like Parkinson is there, Hana? Okay, this is the data for you people, nothing new to you. It's a neurodegenerative disease. How many percent of people are affecting? What is the reason? Pathology involved? Okay, and my target was that I got to know that me and my research partner, Dr. Sangeeta, in my department, she said ki ma'am, NF-kappa receptors are being involved in the pathology of the Parkinson, but still now in the market, though no NF-kappa is there in that. So no doubt NF kappa inhibitors are there, but not in the term of for Parkinson, but they are present for the some another diseases. So what we did, we picked it from Medchem Express. This is again one of the another database, you can write it down. This is free, Medchem Express. It is again target wise, chemical point of views wise, many point of views, it is having number of molecules in its database. So Medchem Express, we used it and we filtered out 6 molecules from that databases. NF kappa that was showing the some good potential. So what we did, we picked the Parkinson disease target. No doubt they are the known NF kappa inhibitors, but they have not been exploited or studied for the Parkinson. So what did we went to the PDB site and we tried to look into all these 6 molecules, and the result is in the form of the D scores, number of residues, type of interactions being involved, and jitna negative mein hota hai, the negative is the score, the better is the molecule. If you look into, you will say ma'am, ursolinic acid at the candidate number, oh sorry, sulfasalazine. You will say what ma'am? Sulfasalazine is showing the best results, okay. -55 followed by -42 and so on ardosine, and I3C is standing at the second number, 4th number, okay. So we are interested in how many type of the residues are being involved. Mesalamine तो है ही नहीं आ रहा, normal alkyl residues आएंगे, hydrogen bonding, hydrophobic. So this was the kind of the basic information we tried to sort it out, okay. Now I told you that what are the 2 tools being used. Primary screening is based upon the docking and secondary screening is based upon the ADME. So we tried to study the pre-ADME. What is pre-ADME? Predictive ADME. Okay, एक होता है ADME. ADME means I am doing it in vitro or using in vivo models, but when there is a pre word, it means I have done it by using predictive data, right. So we tested all these molecules based upon their human intestinal absorption, then plasma protein, blood brain barrier, etc., etc., and we try to combine. If you look into, let me show you the one slide next. X log P. In our drug industry, log P holds very importance. Log P is what? Lipophilicity. Your drug need to have a perfect balance so that it can cross the blood brain barrier and it can reach to the target. If it be too hydrophilic, it is not will be able to cross. If it be too lipophilic, then it will cross the brain also. So it's there is an optimal value of 2 to 5 के बीच में आता है. So they said that now if you look into this score, docking score, here the first molecule was sulfasalazine. Sulfasalazine. If you look into the sulfasalazine structure, it was having carboxylic group, carboxylic group, carboxylic group. It was too polar in nature, not found to be optimal for the study. No doubt it was showing the good. Now what is the difference? How you can say ma'am, the drug is showing the good interaction in the docking, but you are not saying it's good for the, in order to reach to the, in order to show the response at the target site, your drug should get absorbed, it should get distributed, then it should act on the target, after that it should get metabolized or get excreted. Do you think the metabolism before the drug reaches to the target is good or bad? Bad. If the drug is getting metabolized before it reaches to the target, is it good or bad? पहले हो जाएगी. So metabolism need to be studied. ठीक है. So we applied all this filter. No doubt it's showing that sulfasalazine is really 55 score, but when it comes to the X log P कितना आया उसका? 0. So we cancelled out. It's not only the one parameter. Docking is the static studies. Docking is static study. In case you want to support the static binding interactions, you need to do what? आज की term में एक और use होता है term molecular simulations, dynamic. Dynamic क्या होता है? Keep on changing. That person is very dynamic in nature, right? Static means ये receptor है और पड़ा हुआ है. Because I am doing the docking on the computer system, I am not providing you the actual physiological condition what is present in my body. In my body, protein is not present like that. It is continuously contracting, relaxing, contracting, relaxing. So I need to confirm my binding interaction by doing the supporting studies of that is called the dynamic studies, molecular simulation studies. So that time we were not having the molecular simulation software with us, but presently it is with us. So it's not only the docking is statics, but in order to show the interaction at the target site, my molecule should reach from mouth to that side. It means absorption should be good. Absorption कैसे good होगी? जब HI, human intestinal absorption. So we try to understand all these parameter, molecular weight. When you will come into certain market में देखोगे, have you heard about the Lipinski's rule of 5? Very good. What is that? Good. So कहाँ से आया है? Anyone? Who Lipinski क्या है? Scientist का नाम है या क्या है? Lipinski was a scientist. Scientist होने के लिए you need to be psyche at this site, right? So he was a scientist. He observed that there are market में he did research on one topic. He said he took 2000 molecules from the market that are drugs, successful drugs. He studied what is common among these 2000 drugs that make them successful and make them different from the unsuccessful drugs. And he noticed that इतने हाइड्रोजन बॉन्ड डोनर थे, इतने हाइड्रोजन पॉइंट एक्सेप्टर थे, इतना लॉग भी था, इतना मॉलिक्यूलर वेट था. That's why it is called the Lipinski rule of 5. He studied 2000 drugs in detail. What were the common features that make them successful? So that means that's why he said in a drug, a chemical, if you want to become a drug, it should have these features: donor, acceptor, log P, molecular weight and the polar surface area. I hope you got it that. So that is Lipinski rule of 5, powerful rule. उसके बाद बहुत सारे आए, many rules are coming, but no one can beat the Lipinski rule. If it is any candidate is having all these 5 features, that's why a computer can, do you know that if you add your molecule, now after this we will be doing one study. I will give you one assignment. You will put into the ChemDraw. You will try to predict the properties. A computer can predict 240 properties. But what is the logic? So out of those 240 properties, what are 5 important properties I need that? Okay. So try to focus on that. At least it should have these 5 parameters, and after all these, no doubt you would say, ma'am, indole I3C was falling on the 4th number according to the D score. But after applying the parameter of ADME, Lipinski rule of 5 and the molecular dynamic studies, we reached to the I3C as the 4th number candidate. Somehow, some certain time it was the reason one molecule, no doubt it was having good D score, but it was not having good log P value. Another second molecule filtered it out, it was having no doubt good score, but molecular weight was not good. Third molecule that was very costly. Let me admit that I was not in position to afford that much amount of money, so I came to the 4th potential candidate. And this is how, and ultimately when we validated the molecule, we identified in the computers, and then we did the experimental validation in our lab, and we got it published in journal of 19.3. So this is how you can enhance your studies, you can upgrade the level of your studies, and try to slowly incorporate any one feature. Look into the acceptability rate, look into the success rate, look into the curtail of the expenses you can solve. Okay. So this is the one of the article, protective effect of indole 3 NF kappa in experimental paradigm of Parkinson disease in silico and in vivo studies. This is a lab article from our lab. Is that okay? So this is I3C, and we are continuing with the, now I have identified. Again, I ask you, I33, did I synthesize this molecule? No. Did I discover this molecule? No. What I did? Identify. Now I got the skeleton from this study. Now what I am trying to do in my lab, I am trying to modify I3C in my labs. I did the modification, I got even better molecule and we got patent on it. That. So this is how. So don't you think this has fastened up the reaction? Don't you think? So computers are not doing anything. Computers are helping you to fasten up the reaction. They are helping you to make better decisions. They are helping you to reduce the time, reduce the cut on the cost. Okay. I was having many more examples from our lab, but it's okay. This is a collective study from with Dr. Tanzir. That this is no doubt all credit goes to Dr. Tanzir. He gave me a piece of work to do on the computers. I did the computational and synthetic part, all biological part goes to her credit, and we have many papers out of these studies. Thanks to you, ma'am.
Thank you, Dr. Nilima. We will continue this session. We have practical session after a cup of tea. We have a quick break of just 10 minutes. Please be hurried. So that
All I have kept on a छोटी सी एक्सरसाइज करें. Hello. Am I audible to everyone? Is that okay? Open the chem draw, pick any one drug from the board, get its structure from the Google, then try to imagine and look into the different fragments. All the fragments are lying in the library of the chem draw. Try to rejoin them and you will get the one structure. If there is any disturbances in the stereochemistry or in the double bond configuration confirmation, chem draw itself will started flashing out.
Here I am typing A.
Where is the structure now? Shivani, which software you are? अच्छा
ये न्यू डॉक्यूमेंट
हाँ मैं वही इनके देख रही थी ना इसको ये क्यों नहीं फाइंड हो रहा ना
ये मैम इन्होंने अभी हो गया ना मे भी शायद तब आ रहा
नाउ हियर यू नीड टू ड्रॉ लिख इट हियर ठीक है इट्स वेरी सिंपल राइट व्हेन यू पुट अ डबल बॉन्ड सिंगल बॉन्ड राइट यू कीप ऑन राइट इट हार्डली टेक 2 minutes get the structure in front of you get the structure na you need to open a blank document you need to open a blank document get the structure this is a very 0.1% of the work we will be doing today. And I hope आज आप शाम को जाके LinkedIn ज़रूर देखेंगे. Look out for the jobs that are waiting for you people. I stopped miss showing you the slides. 90+ startups have like India में start हुए हैं regarding the use of the computational tools that are inviting you people. 90+ startups around computational tools in the drug discovery. And you need the basic science knowledge. All people knowing that knowledge of the biology, pathways, whatever in it. You just need to take the help of the computer and then you can go in. One dentist, interesting story of dentist is there. One dentist was there and he was passionate about the language, right, computers. After working for 2 to 3 years in the dentist in his clinic, he immediately dropped and presently he is having 700 crore turn up using computational tool in AI. It was day before yesterday news. Dentist, just all you need a passion. Machine is there, simply white collar jobs are there, right? Hanji, are you comfortable? Done? Are you done with the structure? Okay, now select the structure. Select the structure. Select the structure. Have you selected? Okay, go to from file and predict the properties. There must be predict properties. कहीं लिखा होगा. Shivani, come this way. They have drawn the structure and help them to predict the properties. Predict the properties. Drawn. Now select the structure. Select the structure. Stereochemistry is really important. I hope you know the meaning of stereochemistry, right? Below the bridge or up the plane, above the plane. And one of the interesting example present at our place is cetrazine and levocetrazine. Earlier it was in the market cetrazine. Now it is the levocetrazine, because cetrazine, when it was cetrazine, it was having the mixture of the plus and minus both. But when it started showing the side effect, then they tried to separate out the levo isomer only, and now it is in the market levocetrazine more potent in nature. So stereochemistry, keeping the bond above and plane will change all the properties. मेरे specs कहाँ गए? यहाँ पे ये रहा. ओह ये टूल मत यूज़ करो, या ये वाला भी कर सकते हैं, ठीक है ना. नाउ स्ट्रक्चर यहाँ लिखा होगा ना, एटम प्रॉपर्टीज, बॉन्ड प्रॉपर्टीज, ठीक है. लेट मी गेट माय प्रेडिक्ट NMR, IR, ठीक है. कन्वर्ट नेम इनटू स्ट्रक्चर, स्ट्रक्चर टू नेम, देन Smile is, try to get it done. शिवानी शो मेरे को स्पेक्स लेने के आने दो, इनको प्रॉपर्टी करना, मैं स्पेक्स लेके आती हूँ बेटा. स्ट्रक्चर सेलेक्ट करवा दिए, अब स्ट्रक्चर के अंदर जाओगे ना, एटम प्रॉपर्टीज एंड बॉन्ड प्रॉपर्टीज तो आएगी. ठीक है, इसको सेलेक्ट करो. वो स्माइलीज वाला, हाँ स्माइलीज का है ना, स्माइलीज बताओ इनको, स्माइलीज करवाना ना मुझे.
हाँ जी, एडिट कॉपी एज
Smiles. Now I will add here. Can I get my presentation once again, just for 2 minutes? हाँ? आपका लैपटॉप चला गया? नहीं, मेरे पास लैपटॉप नहीं था. अच्छा. I hope you all have converted it into. कोई बात नहीं, it's okay, okay. All of you have check the structure. Got the structure? Now you can predict the atom properties. How many type of the atoms are present, bond property, bond angle, then overall molecular properties, molecular weight, log P, polar surface area, lipophilicity, right? You can predict the NMR, you can predict the IR, and the most important is that convert into.
सिर्फ मैम ने देखनी है अपनी, हाँ उनकी भी
ये मैं देती हूँ. इनको ना स्माइल्स दिखाती हूँ. इन दिस वर्ड देयर आर द 2 वर्ड्स. आप लोग अभी यहाँ पे देखेंगे, एक स्माइल है, एक इंचल की होती है. इंचल की इंटरनेशनल की होती है. एक मॉलिक्यूल को देखने में, चाहे आप यूएस में बैठे हैं, चाहे जापान में, कनाडा में, कहीं पे भी बैठे हो, आईयूपैक नेम सुना है आपने, राइट? इंटरनेशनल यूनियन प्रोप्राइटरी, राइट? नाउ कॉपर सल्फेट इज देयर. हम इंडिया में नीला थोथा बोलते हैं उसको, राइट? कॉपर सल्फेट इज देयर, बट कॉपर सल्फेट इज हैविंग इट्स ओन नेम इटसेल्फ. सिमिलरली इन दिस ग्लोब, chemical is known by 2 things, smiles and इंचल की, and this is called chem informatics. ठीक है, कंप्यूटर की लैंग्वेज में हम इसको केम इन्फॉर्मेटिक्स बोलते हैं. इंचल की INCHLKEY था कि मेरे को दिखा देती है इनको, कोई बात नहीं, अगर आ सकता है तो. नहीं, मेरे को इनको ही दिखाना था. अगर लग सकता है कंफर्टेबली, इनको ही दिखाना था कि स्माइली और इंचल की कैसे दिखती है. ऑल यू गॉट द स्माइल्स ऑफ योर स्ट्रक्चर. स्माइल नहीं करना आया. सिलेक्ट द स्ट्रक्चर, कन्वर्ट इनटू स्माइल्स लिखा होगा. जब आप कल को कंपटीशन वर्ड में जाएंगे तो बहुत सारी लैंग्वेजेस होती हैं, बहुत सारे टूल्स हैं. मेरे पास वो ना लेक्चर ही नहीं है, वो बोला कैम इन्फॉर्मेटिक में अपने आप में इतनी स्लाइड्स है ना कि एक सॉफ्टवेयर दूसरे सॉफ्टवेयर से कैसे वेरी करता है. उसमें जैसे लाइक लेट इट से इफ आई एम हैविंग वन लैंग्वेज, मतलब इफ आई एम हैविंग वन स्ट्रक्चर, इट्स नॉट नेसेसरी. जैसे मान लो ये ऑटोडॉक पे काम करना चाहते हैं, ऑटोडॉक फाइल इज नॉट कंपेटेबल विद द स्ट्रक्चर. सेम वे मैनी सॉफ्टवेयर आर देयर, बट इफ यू हैव स्माइल ऑफ दैट एनी स्ट्रक्चर, इट वुड ओपन फॉर एवरीवन, देन नो नीड टू वरी अबाउट इट. वो बताते नहीं कई लोग. कंपैटिबिलिटी होती है.
कन्वर्ट करते थे क्या उसके कंपैटिबल.
हाँ, कंपैटिबिलिटी का बड़ा होता है.
कहीं पे POLG होता था, क्या क्या अलग अलग फॉर्मेट्स.
हाँ जी, स्माइल्स मिल गया बेटे आपको. लास्ट बेंचेस क्या स्माइल ऐसे आएगा, कार्बन HO, सिंपल. कन्वर्ट करके स्ट्रक्चर में कन्वर्ट करके नहीं है.
स्ट्रक्चर बन जाता था, वो ऑप्शन किधर आता है? ये हमने सेलेक्ट किया.
हाँ, यहाँ जाओ. मैं लेने स्पेक्स गई थी. खोलो इसको, व्यू. ओहो, नहीं, इसको खोलो. किस में था ना, मैं ऐड स्पेक्स, लेने स्पेक्स जाती हूँ. हाँ, एडिट करके वहाँ पे. हाँ, ये देखो, कॉपी एज, कॉपी पेस्ट स्पेशल. ये रहा स्माइल्स, करो. ये रहा इंचल की भी आएगी यहाँ पे. इसी की स्माइल भी निकालो, इसी की इंचल की भी निकालो. आएगा, पेस्ट करो इसको. आया नहीं? हाँ करो. नहीं आया क्यों नहीं? पेस्ट करो. आपका स्माइल आ गया? स्माइल आया नहीं आपका? ऐड कंट्रोल प्लस, हाँ ऐड कंट्रोल प्लस स्माइल आना चाहिए बेटे, नहीं तो कॉपी रह जाएगा ना। ओहो सॉरी, स्ट्रक्चर सेलेक्ट करो पहले, एट ए टाइम एक ही कर लो ना, उसको तो डिलीट कर दो। सिर्फ इसको सिलेक्ट करो, ऊपर वाली को। हाँ स्लोली, ठीक है। नाउ गो टू एडिट, एडिट, हाँ स्माइल, इसको करो ना, ऐड कंट्रोल प्लस सी करो ना, कमांड यूज़ करो ना बेटा, ऐड कंट्रोल प्लस सी। मैं स्पेक्स लेके आया, अब देखो आया पेस्ट स्पेशल। अब एडिट में फिर से जाओ, पेस्ट स्पेशल में आओ, स्माइल्स करो, कंट्रोल ऑल्ट कंट्रोल प्लस एफ लिखा था ना उसने, पहले क्या था, ऑल्ट कंट्रोल सी, नाउ ऑल्ट कंट्रोल पी। चेक करो, शिवानी इनका वो नहीं आ रहा, स्माइल नहीं आ रहा, करो स्माइल क्यों नहीं आ रहा? अच्छा सबको स्माइल आ गया, आपका आ गया स्माइल? क्या है आपकी, कौन सी स्ट्रक्चर है? सेलिग्लिन, सेकेंड वन, दिस यू कैन यूज डिफरेंट टूल्स, ये फ्री वाले हैं। इफ यू वांट, इफ एनीवन वांट टू हैव अ जस्ट आइडिया, in case if any one of this tool is helpful to you, you can get a prick on it. Some of the tools I have put it on the board, rest you can do it. आप गूगल करना शुरू करेंगे, it will help you out. Some of the different tools, toxicity prediction, protein ligand binding interactions, molecular predictions. हाँ बेटा, इसमें नहीं है मेरे बेटा, आई विल टेल यू द सॉफ्टवेयर, अभी बता रहे हैं, वी आर कमिंग टू दैट टूल आल्सो। अभी यहाँ से सिर्फ आपने, कैन आई हैव अटेंशन योर प्लीज, फ्रॉम दिस कैम ड्रॉ you got the structure and you got the smile. Smile is universal language that flowed from one system to another, from one language to the another. Okay, sometimes compatibility issues are there, but if you have the smile of any structure, you can take it to the any another platform, right? Now from this you have got the smile. Now कौन सा tool हम Swiss ADME, go to the Swiss ADME tool, Google it out, Swiss ADME tool. I think it might be written Swiss ADME, and here I was showing you that, look at this startups. In 2023, 11.78 billion USD in India country GDP is going to be 1 trillion by 2029. So these all are working on only AI tools in the predictive analysis, imaging, diagnostic, hospital, remote, drug discovery, and this is one of the era that is waiting for you all. जब आप कहीं पे फाइल देखते हो ना, whenever you will be using, आपने कहीं सुना होगा MOL, SDF, SMILE format. What is the full form of the SMILE? It is a simplified molecular input line entry. Line. You are getting the SMILEs in the form of a line, right? Simple line entry. That's why it is called as SMILEs. Okay, system line notation for a molecule. आपने molecule कैसे draw किया? Benzene, then structure, but the same molecule you have getting in the form of a line. So that is the simple line notification or a representation of a given molecule. Okay ji, that is called the smiles. Whenever in the computer world try to get the initial key, this is basically आ गया इंचल की, बहुत सारा है. इंचल की कम्स इन द लेयर्स, 4 लेयर्स में आता है. इट इज A B C L M N टाइप्स आ रहा होगा, राइट. उसमें सिंपल कार्बन हाइड्रोजन स्माइल्स में आ रहा होगा, राइट. आपके लिए ये जॉब भी वेट कर रही है, एक स्ट्रक्चर को स्माइल में कन्वर्ट करना या स्माइल से प्रेडिक्ट करने में कि कौन रहा है. हम लोग ही तो कर रहे हैं. हम जिसे कहते हैं जॉब्स वेट कर रही हैं, वो जॉब्स यही तो हैं, राइट. जॉब्स डाटा एंटर करने में भी अपने आप में इंफॉर्मेशन जॉब है, उसमें से निकालने में भी. सो दीज आर डिफरेंट काइंड ऑफ द जॉब्स. इफ यू आर, अब हम लाइक इफ समवन इज वर्किंग एट द Schrödinger, राइट, नाउ ही इज प्रोवाइडिंग द टेक्निकल सोल्यूशन टू अस. दे आर नॉट नथिंग, दे आर नॉट कोई सेपरेट पीपल नहीं है, दे आर नॉट डिफरेंट पीपल, दे आर बायोकेमिस्ट, बायोलॉजिस्ट पीपल, फार्मासिस्ट हैं, हमारे जैसे ही लोग वहाँ बैठे हैं. उनके लिए सिर्फ कंप्यूटर है, लेकिन कंप्यूटर के ऊपर काम किसको आना चाहिए, जब तुम्हारे पास नॉलेज है. ठीक है. सो दिस इज द क्रक्स ऑफ द टुडे लेक्चर बेसिकली. इफ यू लुक इनटू द SBD इज द स्ट्रक्चर बेस्ड ड्रग डिजाइनिंग, डायनेमिक करना, डॉकिंग करना, होमोलॉगिंग मॉनिटरिंग करना, डाटाबेसेस मैंने आपको टर्म बता दी. राइट हैंड साइड आई हैव नॉट कवर्ड बिकॉज दैट इज टोटली केमिस्ट्री पार्ट. दैट वुड आई थिंक बिगनिंग अभी आपके लिए, अदरवाइज यू मट हैव गॉट बोर्ड. तो मैंने इस साइड को कवर नहीं किया, मैंने लेफ्ट हैंड साइड को टच किया. ओके, अदरवाइज दिस ऑल टूल्स आर वेटिंग फॉर यू. एंड वहाँ फिगर में देखोगे डाटाबेसेस, ये सारे डाटाबेसेस हैं. ड्रग बैंक, अब एक मैं आपको एक डाटाबेसेस मतलब समझाती हूँ. ड्रग बैंक का डाटाबेस और एक केमिकल डाटाबेस. एनीवन कैन हेल्प मी द अंडरस्टैंडिंग द डिफरेंस? मैंने हिंट दे दिया. केमिकल डाटाबेस और ड्रग बैंक का डाटाबेस. वेरी गुड, शाबाश. केमिकल डाटाबेस है जिंक, नीचे है ड्रग बैंक का डाटाबेस. यस, केमिकल डाटाबेस में वट एवर द मॉलिक्यूल, वेदर इट इज शोइंग थेरापेटिक एफिकेसी और नॉट, इट हैज बीन सिंथेसाइज्ड बाय मी, इट्स प्रॉपर्टीज आर पूरी डिटरमाइंड और नॉट, इट इज एनी काइंड ऑफ मॉलिक्यूल. आज मैंने सिंथेसाइज किया, मैंने पब्लिश कर दिया, टुमारो इट विल बी इन द डाटाबेस. ड्रग बैंक इज होल्डिंग द इंफॉर्मेशन ओनली द अप्रूव्ड ड्रग्स. ओके, databases are both, but so other databases are ChemSpider, ChemBill, GDP, COD, many more databases are there for you all. So initial key, you got it? Initial key आ गई. So have you noticed the difference between the smiley and the initial key, right? एक initial key, एक molecule है, I am having prostaglandin का नाम सुना है सबने? Prostaglandin का initial key 4 पेजेस में जाता है. Now if I ask you to convert the prostaglandin initial key 4 पेजेस में लिखाने के लिए, आपको शायद नहीं याद रहेगा, but this much of the knowledge is being stored in the computers, जो हमें सालों साल लग जाएंगे. Is that okay? Initial key और smile में क्या है? Smile, it is a single line. Initial key is holding the information in 1, 2, 3, 4 layers. It is holding the information about the stereochemistry. It is beyond my approach. It is beyond my cup of tea. It's not my cup of tea. I cannot understand the initial key. आप कहीं पे भी गूगल आउट करोगे, स्माइल समझना आसान है. Initial key is beyond human tendency capacity. It is beyond. You won't be able to understand. Yes, 4 layers क्या बताती हैं? मॉलिक्यूलर फॉर्मूला बताती हैं, उसकी स्टीरियोकेमिस्ट्री बताती हैं, देन उसके सेंटर्स बताते हैं, मेनी मोर इन्फॉर्मेशन इज दिस बीइंग स्टोर्ड. ओके, सो दैट इज बियॉन्ड, वो मशीन लैंग्वेज है ज़्यादातर. स्माइल्स इज सिंपल लैंग्वेज. दैट्स वाई जो रोटेट करती है वो लैंग्वेज है स्माइल्स है. अब आपने स्माइल आ गया आपको. नाउ हैव यू ओपन द स्विस ADME टूल? स्विस ADME टूल खोल लिया? मानसी कह रही हूँ, शिवानी खुलवा दिया स्विस ADME? Now get this Swiss ADME smile into the Swiss ADME. Get this smile into the Swiss ADME tool and try to Swiss है Swiss का, ठीक है, कुछ नहीं है, मतलब सबसे पहले वहाँ से हुआ था ADME tool. and try to predict the properties like absorption, distribution, metabolism and excretion of this given drug to you. बस निकालो, मेरे पास है नहीं है. So this was like I have already. हाँ
स्लाइड दिखानी कुछ?
हाँ दिखाओ, नहीं बस कर लो, निकाल लो. चलो हाँ जी, विच साइड? आप लोग किस साइड पे काम कर रहे हैं? प्री एडेमी खुल गया, ठीक है. सो वहाँ पे स्ट्रक्चर डालो अपना. Swiss ADME and pre-ADME side, though both sides they work differently. One accept the smiles and one accept the structure. इसी तरह से number of the softwares are there. Every software, why we are having number of the different type of mobiles? Every mobile is having its own unique characters. खुल गया? Try to predict the properties. Going fine? What's an issue? दोबारा हाँ. Now again one more observation. जैसे जैसे आप काम करते जाओगे, pre-ADME I think it is not accepting the structure that have been drawn in the ChemDraw. You need to draw afresh in the pre-ADME tool itself, right? So this is called compatibility. Pre-ADME is not accepting the MoleSoft file that you people have drawn in the ChemDraw. You need to draw afresh in the pre-ADME tool. Many properties are there. You can predict the toxicity. नेट डायरेक्टली सर्वर से है ना. नाउ आई होप अगेन इट्स वन छोटी छोटी ऑब्जर्वेशन है. दीज आर ऑनलाइन सर्वर टूल्स, राइट. एक होता है फ्री सॉफ्टवेयर कि आपने डाउनलोड करके अपने मोबाइल पे रख लिया, बट हेयर यू पीपल आर वर्किंग डायरेक्टली ऑन द सर्वर. ओके, so if you are working for some novel compound, don't do the mistake by predicting the properties over here, because this way you will choose the chance of novelty and so the patentability chances. You need to be very cautious. These are small observations. Okay, saying if you are just learning the things, it's okay, but if you are going to plan something novel out of it, then because once the molecule is in air, you lose the novelty. So don't go for that. बस उसको सिलेक्ट करो, प्रॉपर्टी प्रेडिक्ट करो, सिलेक्ट द स्ट्रक्चर एंड वहाँ पे ऊपर जाइए एंड प्रेडिक्ट द प्रॉपर्टीज, ADME प्रॉपर्टीज. Have you selected the structure? Second bench. Shivani, second bench. Pre-ADME tool में they have drawn the structure. Now let them select the structure and predict the properties. हाँ जी, at which stage you are? What? We submitted for the property prediction. It's running. Executed. Or you got the properties also. What type of properties you have got? Okay. Solubility, molecular weight. Huh? CSA. Okay. Caco permeability. हाँ. Enzymes में. How many enzymes are there in my body? 70 enzymes are there. If I want to study one enzyme of one particular drug, right? I am giving a drug to my body, right? My body drug need to go through the saliva, then the fecal, stomach, small intestine, large intestine, and number of the organs are coming on the way. It may get metabolized before I want my drug to reach to the target. It should not get before in advance. It should get post. So it would be studied. Your computer got the power to study its metabolic fate by all these 70 enzymes, right? So it will help you to understand if the drug is getting, if the possible जो भी आपने नया एक molecule बनाया है, if it is getting metabolized in advance, why I would be putting it for the animals, right? So it means all the 70 molecules. What I will do, if you remember my machine learning model, I given that this is my molecule, physicochemical data. I have given the organ kidney. It will select kidney के अंदर कौन कौन से एंजाइम है. It will determine the model बन जाएगा and then you will predict the property कि आपका molecule मेटाबोलाइट होगा या नहीं होगा, 70% होगा, 10% होगा, कितने मेटाबोलाइट्स बनेंगे, वो मेटाबोलाइट टॉक्सिक हैं, सेफ है, उनका कार्सिनोजेनिक इफेक्ट होगा. Everything will be done. You done the properties? What kind of properties? Okay. Now what is the blood-brain barrier or range you are getting? 0.8, है ना? So now it is the these are the CNS almost neurodegenerative. You want the drugs to get absorbed or not? For CNS drug you want to get absorbed. So these now, if you have let it say you people are working on the Swiss ADME and he is working on the pre-ADME tool and I am working on the Swiss Schrodinger. So every software would have its own optimal values. Don't say I am getting 0.88 day and it would be having different value. So ये गलती मत करना, ठीक है ना? Every tool would be giving its own. उनके tutorial में जाएंगे, it will give you that if your compound is falling within this range, this range, this range, this range, it is okay, it is safe, it is toxic, it is metabolizing, everything is there. So you need to cross check with the tutorial observations, वहाँ पे लिखा होगा. So these are the software. Now from the given small exercise, you have noticed that you have learned SMILES, you have learned Intel, you have learned pre-ADME tool, Swiss ADME SMILES ले रहा है, pre-ADME tool structure, ChemDraw में बनाई हुई structure नहीं उठा रहा, ना वो SMILE उठा रहा है, लेकिन वो क्या कर रहा है, वो खुद की structure को अपना बनाने में कर रहा है, ठीक है? So you may get अब ADME आपकी कितनी ड्रग, उसका कितना आया ब्लड ब्रेन बैरियर? 1.98, उनका 0.88 आ रहा है. और अगर मैं इसको क्रॉस अब कोशिश करना, आज ये स्मॉल एक्सरसाइज है, ट्राई टू correlate this predictive data with the, because these are the already known drugs na, so try to correlate this predictive data with the already reported data and try to establish in silico in vitro correlation कि वेदर कितना प्रिडिक्टेबिलिटी है. देखो वेदर फॉरकास्ट के लिए भी सॉफ्टवेयर्स हैं हमारे मोबाइल में, सबके पास हैं, गूगल भी है ना, हम सब कहते हैं, एप्पल का अपना वर्जन आता है, एप्पल डज नॉट अलाउ टू एंटर एनी अनदर ऐप, उसके अपने ही सॉफ्टवेयर्स होते हैं. तो सिमिलरली सम टाइम मैं ये नहीं कहती कि सॉफ्टवेयर्स आर 100% राइट, नो, नीदर सॉफ्टवेयर्स नीदर कॉम्पिटिशनल टूल्स कैन ओवरपावर और रिप्लेस द ह्यूमंस, ह्यूमन इंटरवेंशंस ऑलवेज बीइंग रिक्वायर्ड. अगर मैं वेदर फॉरकास्ट की प्रिडिक्शन करना चाहती हूँ तो सॉफ्टवेयर में तो मैं ही डेवलप करूँगी ना, राइट. अब उसकी कितनी एक्यूरेसी कितनी होगी, कितने मैं स्टैट्स लगाती हूँ, कितना एल्गोरिदम लगाती हूँ. आई होप यू माइट हैव हर्ड अबाउट न्यूरोनल नेटवर्क, हम करते हैं ना. तो न्यूरोनल नेटवर्क क्या होता है, जिस तरह से नेटवर्क, जिस तरह से हमारे ब्रेन के अंदर डेंड्राइट्स का इतना नेटवर्क है, सेम वे कंप्यूटर्स के अंदर इतनी इंफॉर्मेशन है. यू पीपल कैन ट्राई द न्यूरो नेटवर्किंग फार्माकोलॉजी. व्हाट इज नेटवर्किंग फार्माकोलॉजी? एनी आइडिया? Yes, number of the genes over there, number of the diseases are there, try to apply, and in order to do this the same studies it will take number of the years. ठीक है. और वन इंटरेस्टिंग पैरामीटर मैं हमेशा शेयर करती हूँ, बनाना और तुम्हारे में कितना परसेंट जिनोम सेम है? हाँ, 70% हम बनाना के साथ शेयर करते हैं. वो बनाना बन गया, हम ह्यूमन बन गए, राइट. सो वेरी इंटरेस्टिंग थिंग्स आर पेंडिंग. अभी तो 1% भी नहीं, 1% तो बहुत दूर की बात है. हमने वी हैव डन जस्ट स्मॉल पोर्शन ऑफ इट. सो लिंक्डइन पे जाइए, जॉब्स एक्सप्लोर कीजिए, स्टार्टअप्स देखिए, गूगल आउट कीजिए, चैट जीपीटी से पूछो, परप्लेक्सिटी से पूछो, जो जो भी टूल्स तुम लोग यूज़ करते हो, एंड ट्राई टू मेक इट इंटरेस्टिंग. सो आई होप यू हैव ऑल डन द प्रॉपर्टी प्रेडिक्शंस. ट्राई टू कोरिलेट दिस डाटा, प्रेडिक्टिव डाटा, दिस इज कॉल्ड predictive data, okay, and then correlate with the actual data and then try to you can comment upon the reliability of the software कि ये कितना ठीक है. अब ये फ्री सॉफ्टवेयर है. मैंने आपको यहाँ पे एक छोटी सी ऑब्जर्वेशन और बताई है कि if you are working on some novel molecules, don't do the mistake by putting it on the air. Once it is in the air, then you lose the novelty, okay. In such cases always go for commercial license. Thank you so much.